All right, it's 12:01, so we'll go ahead and get started. So on behalf of the co-chairs and the NIMH Planning Committee, I'd like to welcome you back to day 2 of the NIMH Placebo Workshop, Translational Research Domains and Key Questions. Before we begin, I'll just go over our housekeeping items again. So attendees have been entered into the workshop in listen-only mode with cameras disabled. You can submit your questions via the Q&A box at any time during the presentation, and be sure to address your question to the speaker that you'd like to respond. For more information on today's speakers, their biographies can be found on the event registration website. If you have technical difficulties hearing or viewing the workshop, please note these in the Q&A box and our technicians will work to fix the problem. And you can also send an email to nimh@mn-e.com and we'll put that email address in the chat box for you. This workshop will be recorded and posted to the NIMH event webpage for later viewing. Now I'd like to turn it over to our workshop co-chair, Dr. Christina Kusin, for today's introduction.
Thank you so much, Erin. Welcome, everybody. It's very exciting to be here for this event. My job is to provide you a brief recap of day 1 and to introduce you to the speakers of day 2. Let me share my slides. Again, thank you to the amazing planning committee. Thanks to their effort, we think this is going to be a success. I learned a lot of new information and a lot of ideas for research proposals and research projects from day 1. Very briefly, please go and watch the videos. They're going to be uploaded in a couple of weeks if you missed them, but we had an introduction from Tor, my co-chair. We had an historic perspective on clinical trials from the industry regulatory perspective. We had the current state from the FDA on placebo. We had an overview of how hard it is to provide the right sham for device-based trials and the challenges for TMS. We've seen some new data on the current state of placebo in psychosocial trials and what is the equivalent of a placebo pill for psychosocial trials, and some social neuroscience approach to placebo and analgesia. So we've come a long way from snake oil, and we're trying to figure out what is placebo. Tor, my co-chair, presented some data on the neurocircuitry, underlying placebo effect, and the questions how placebo is a mixture of different elements, including regression to the mean, sampling bias, selective attrition for human studies, the natural history of illness, the placebo effect per se that can be related to expectations, context, learning, and interpretation. We have seen a little bit of how is the impact on clinical trial design and how do we know that something, it really works or whatever this it is. And why do even placebo effect exist? It's fascinating ideas that placebo exists as a predictive control to anticipate threats and the opportunity to respond in advance and to provide causal inference, a construct perception to infer the underlying state of body and the world. We've seen the historical perspective, and Mia Kin and Mike Detke provided some overview of 25 years of randomized controlled trials from the data mining in major depressive disorder, schizophrenia trials, and the lessons we have learned. We have seen some strategies, both historical strategies and novel strategies to decrease placebo response in clinical trials and the results. Start from trial designs, SPCD, lead-in, placebo phase, and flexible dosing, use of different scales, the use of statistical approaches like last observation carried forward or MMRM, centralized ratings, self-rating, computer rating for different assessments, and more issues in clinical trials related to patient selection and professional patients. Last but not least, the dream of finding biomarkers for psychiatric conditions and tying clinical response to biomarkers. And we've seen how difficult it is to compare more recent studies with studies that were started in the 90s. We have the FDA perspective with Tiffany Farchione, and this placebo being a huge issue from the FDA, especially the discussion towards the end of the day was on how to blind psychedelics. We've seen an increase in placebo response rate in randomized controlled trials, also in adolescent studies, and considerations from the FDA of novel design models in collaboration with industry. We had examples of drugs approved for other disorders, not psychiatric condition, and made me realize how little we know about the true pathophysiology of psychiatric disorders that are likely also heterogeneous conditions. And that made me very jealous of other fields because they have objective measures. They have biology, they have histology, they have imaging, they have lab values, while we are far behind and we are not really able to explain to our patients why our medications are supposed to work or how they really work. We heard from Holly Lisenby and Judith Deng, the sham, the difficulty in producing the right sham for each type of device, because most of them have ancillary effects that are separate from the clinical effect, like the noise or the scalp stimulation for TMS. And it's critical to obtain a true blinding and separating sham from verum. We have seen how in clinical trials for devices, expectancy from the patient, high-tech environment, and prolonged contact with clinicians or staff may play a role. And we see how difficult it is to develop the best possible sham for TMS studies and TDCS. It's really complicated, and it's so difficult also to compare published studies in meta-analysis because they've used very different type of sham. Not all sham are created equal, and some of them could have been biologically active, so therefore invalidating the result or making the study uninformative. Then we moved on to another fascinating topic with Dr. Reif and Dr. Atlas. What is the impact of psychological factors when you're studying a psychological intervention? Expectations, specific or non-specific factors in clinical trials, and what is the interaction between those factors? More, we learn about the potential nocebo effect of standard medical care or being on a wait list versus being in the active arm of a psychotherapy trial. And the fact that we're not accurately measuring the side effect of psychotherapy trial itself. And we heard more a fascinating talk about the neurocircuitry mediating placebo effect, salience, affective value, cognitive control, and how perception of provider, perception of his or her warmth and competence, and other social factors can affect response and placebo response, and the bias in evaluation of acute pain of others, another very interesting field of study. From a clinician perspective, this is, and from someone who conducts clinical trials, all this was extremely informative because in many cases, in our patient situation, no matter how good the treatment is, they have severe psychosocial stressors. They have difficulty to accessing food, to access treatment, transportation, or they live in an extremely stressful environment. So to disentangle other psychosocial factors from the treatment, from the biology, is going to be critical to figure out how to treat best our patients. And there's so much more work to do. Each of us approach the placebo topic for research from a different perspective, and like the blind man trying to understand what is an elephant, we have to integrate, we have to talk to each other, we have to collaborate and understand better the underlying biology, understand different aspects of the placebo phenomenon. And this leads us to the overview for day 2. We are going to hear more about other topics that are they're so exciting: the placebo, the nocebo effect, and other predictive factors in laboratory setting. We're going to hear about genetic of the placebo response in clinical trials, more physiological and psychological and neural mechanism for analgesia. And after a brief break, around 1:30, we're going to hear about novel biological and behavioral approaches for the placebo effect. We're going to hear about brain mapping. We're going to hear about other findings from imaging. And we're going to hear about preclinical modeling. There were some questions yesterday about animal models of placebo. And last but not least, please stay around because in the panel discussion, we are going to tackle some of your questions, and we are going to have 2 wonderful moderators, Ted Kapchuk and Matt Rudorfer. So please stay with us and ask questions. We love to see more challenges for our speakers, and we're going to be, all the panelists from yesterday, from today, are going to be present. Thank you so much. Now, we're going to move on to our first speaker of the day. if I'm correct, according to the last, Luana?
Thank you very much, Christina. First, I would love to thank the organizer. This is a very exciting opportunity to raise awareness about this important phenomenon for clinical trials and clinical practice. And today, I wish to give you a very brief overview of the psychoneurobiological mechanism of placebo and nocebo, the description of some pharmacological studies, and a little bit of information on social learning. That is a topic that has been mentioned a little bit yesterday. And finally, the translational part. Can we translate what we learn from mechanistic approach to placebo and nocebo in terms of disease, etiology, and eventually predictors, the big question. So we learned yesterday that placebo effects are generated by verbal suggestion. This medication has strong antidepressant effects. Therapeutic prior experience, merely taking a medication weeks, days before, begin substitute with a simulation, a placebo, a sham treatment. Observation of a benefit in other people, contextual and treatment cue, and interpersonal interactions. Especially in the fields of pain, where we can simulate nociception, painful experience in laboratory setting, we learn a lot about the modulation related to placebo. In particular, expectation can provide a reduction and activation of part of the brain like frontal area, nuclear accumbens, ventral striatum, and this kind of mechanism can generate a descending stimulation to make the painful nociceptive stimulus less intense. The experience of analgesia at the level of a pain mechanism translates into a modulation, reduction of pain intensity, but most important, pain unpleasantness and affective component of pain. I will show today some information about psychological factor, demographic factor, as well as genetic factors that can be predictive of placebo effects in the context of pain. On the other hand, a growing interest is related to nocebo effects, the negative countersides of this phenomenon. When we talk about nocebo effects, we refer to increase and worsening of outcome and symptoms related to negative expectation, prior negative therapeutic experience, observing a negative outcome in others, or even mass psychogenic modeling, such as some nocebo-related response during the pandemic, treatment leaflets, the description of all side effects related to a medication, patient-clinician communication, the informed consent, where we list all the side effects of a procedure of medication, as well as contextual cues and clinical encounters. And importantly, internal factors like emotion, mood, maladaptive cognitive appraisal, negative valence, personality traits, somatosensory features, and omics can be predictive of negative worsening of symptom and outcome related to placebo and nocebo effects. In terms of nocebo, very briefly, there is a lot of attention again related to brain imaging with beautiful data show that the brainstem, the spinal cord, the hippocampus play a critical role during nocebo hyperagesic effects. And importantly, we learn about placebo and nocebo through different approach, including brain imaging, as we saw yesterday, but also pharmacological approach. We start realizing that placebo effects are really neurobiological effects with the use of agonist or antagonist. In other words, we can use a drug to mimic the action of that drug when we replace the drug with a saline solution, for example. In the cartoon here, you can see a brief pharmacological conditioning with apomorphine. Apomorphine is a dopamine agonist, and after 3 days of administration, apomorphine was replaced with saline solution in the intraoperative room. The goal was to understand if we can mimic at the level of neuronal response the effects of apomorphine. So in brief, these are patients undergoing subthalamic region installation of deep brain stimulation. You can see here a needle reaching the subthalamic nucleus. So after crossing the thalamus, the zona incerta, the STN, and the substantia nigra, the surgeon localized the area of stimulation. Because we have two subthalamic nuclei, we can use one as control and the other one as target to study, in this case, the effects of saline solution given after 3 days of apomorphine. What we found was that in those people who respond, there was consistency in reduction of clinical symptoms. As you can see here, the UPDRS, a common scale to measure rigidity in Parkinson, the frequency of discharge at the level of neurons, and the self-perception. Patients refer sentences like, I feel like after levodopa, I feel good. This feeling good translates in less rigidity, less tremor in the surgical room. On the other hand, some participants didn't respond. And consistently, we found no clinical improvement, no difference in frequency of discharge at the level of a single unit, and no self-perception of a benefit. This kind of effects start to trigger the questions, what is the reason why some people respond to placebo and pharmacological conditioning and some other people don't? I will try to address this question in the second part of my talk. On the other hand, we learn a lot about the endogenous modulation of pain and true placebo effects by using, in this case, an antagonist. The goal in this experiment was to create a painful sensation through a tourniquet. Week 1, we had no treatment. Week 2, we pre-inject health participant with morphine. Week 3, the same morphine. And week 4, we replace morphine with placebo. And you can see that a placebo increased the pain tolerance in terms of minutes. And this was not a carryover effect. In fact, the control at week 5 show no differences. Part of the participants were pre-injected with an antagonist, naloxone, that when we use naloxone at high dose, we can block mu-opioids, delta, and K receptors. You can see that by pre-injecting naloxone, there is a blockage of placebo analgesia. And I would say this morphine-like effects related to placebo given after morphine. We start to then consider this phenomenon. Is this a way for tapering opioids? And we call this sort of drug-like effects as dose-extending placebo. The idea is that if we use a pharmacological treatment, morphine, apomorphine, as I showed to you, and then we replace the treatment with a placebo, we can create a pharmacological memory, and this can translate into a clinical benefit. Therefore, the dose-extending placebo can be used to extend the benefit of the drug, but also to reduce side effects related to that drug. In particular, for placebo given after morphine, you can see in this graph, the effects is similarly strong if we do the repetition of morphine 1 day apart or 1 week apart. Interestingly, this is the best model to be used in animal research. Here at the University of Maryland, in collaboration with Todd Gold, we create a model of anhedonia in mice, and we condition animals with ketamine. The goal was to replace ketamine with a placebo. There are several controls, as you can see, but what is important for us, we condition animals with ketamine week 1, 3, and 5, and then we substitute ketamine with saline along with the CS. The condition at the stimulus was a blue light. And we want to compare this with an injection of ketamine given at week 7. So as you can see here, of course, ketamine was inducing a benefit as compared to saline and ketamine. But what is interesting, when we compare ketamine on week 7 with saline replacing ketamine, we found no difference, suggesting that even in animals, in mice, we were able to create a drug-related effects. In this case, a ketamine antidepressant like placebo effects. These effects also had dimorphic effects in the sense that we observe this in males but not in females. So another approach to use agonist, like I mentioned for apomorphine in Parkinson patients, was to use vasopressin and oxytocin to increase placebo effects. In this case, we use verbal suggestion that in our experience, especially with health participants, tend to create very small sample size in terms of placebo analgesic effects. So we knew that from the literature that there is a dimorphic effects for this hormone. So we inject people with intranasal vasopressin, saline, oxytocin in low dose, and no treatment. You can see there was a drug effects in women, whereby vasopressin boosts placebo analgesic effects, but not in men, where yet we found many effects of the manipulation, but not drug effects. Importantly, vasopressin affects dispositional anxiety as well as cortisol. And there is a negative correlation between anxiety and cortisol in relationship to vasopressin-induced placebo analgesia. A dilemma was, can we use medication to study placebo in a laboratory setting, or can we study placebo and nocebo without any medication? One example is to use a manipulation of the intensity of painful stimulations. We use a thermal stimulation tailored at 3 different levels, 80 out of 100 with a visual analog scale, 50 or 20, as you can see from the thermometer. We also combine the level of pain with a phase to further emphasize these 3 levels of pain. And participants will see an anticipatory cue just before the thermal stimulation. 10 seconds of thermal stimulation to provide the experience of analgesia with the green and hyperalgesia with the red as compared to the control, the yellow condition. Therefore, the next day we move in the fMRI, and the goal was to try to understand to what extent expectations are relevant in placebo and nocebo effects. We mismatch what they anticipate and they learn the day before, but also you can see we tailor the intensity at the same identical level, 50 for each participant. We found that when expectation matched the level of cues, anticipatory cue and phase, we found a strong nocebo effects and placebo effects. You can see in red that despite the level of pain were identical, they perceive red-related stimuli as higher in terms of intensity and green-related stimuli as lower when compared to the control. By mismatching what they expect with what they saw, we block completely placebo effects and still nocebo persists. So then I show to you that we can use conditioning in animals and in humans to create placebo effects, but also verbal suggestion, the example of vasopressin. Another important model to study placebo effects in laboratory setting is social observation. We see something in other people, we are not told what we are seeing, and we don't experience the thermal stimulation. That is the setting. A demonstrator receiving painful, no painful stimulation, and someone observing this simulation. When we test the observers, you can see the level of pain were tailored at the same identical intensity, and these were the effects. In 2009, when we first launched this line of research, this was quite surprising. We didn't anticipate that merely observing someone else could have boosted expectations and probably creating this long-lasting analgesic effects. This drove our attention to the brain mechanism, what is so important during this transfer of placebo analgesia. So we scanned participants when they were observing a video this time, and a demonstrator receiving control and placebo cream. We counterbalance the color, we control for many variables. So during the observation, of another person when they were not stimulated, they didn't receive the cream, there is an activation of the left and right temporoparial junction and a different activation of the amygdala with the two creams. And importantly, an activation of the periaqueductal gray that I show to you is critical in modulating placebo analgesia. Afterwards, we put the placebo creams with the two different colors. We tailored the level of pain at the identical level of intensity, and we saw how, placebo effects through observation are generated. They create strong different expectation, anxiety, and importantly, we found that the functional connectivity between the dorsolateral prefrontal cortex and the temporoparietal junction that was active during the observation mediate the behavioral results, suggesting that there is a mechanism here that may be relevant to exploit in clinical trials and clinical practice. From this, I wish to switch to a more translational approach. Can we replicate these results observed in health participants for nociception in people suffering from chronic pain? So we choose as population orofacial pain, that is an orphan disease. There is no consensus on how to treat it, but also it affects the youngest, including children. So participants were coming to the lab, and as you can see, we use the same identical thermal stimulation, a sham electrodes, the same conditioning that I showed to you. We measure expectation before and after the manipulation. The very first question was, can we achieve similar magnitude distribution of placebo analgesia in people suffering chronically from pain and comorbidities? And you can see that we found no difference between temporoparietal, between the TMD and controls. Also, we observed that some people responded to the placebo manipulation with hyperalgesia. We call this nocebo effect. Importantly, these effects are less prevalent than the benefit that sometimes can be extremely strong, show that both health control and TMD. Because we run experiments in a very beautiful ecological environment where we are diverse, the lab, the experimenters, as well as the population we recruit in the lab has a very good distribution of race ethnicity. So the very first question was, we need to control for this factor. And this turned out to be a beautiful model to study race ethnicity in the lab. So when chronic pain patients were studied by same experimental race, dark blue, we observe a larger placebo effect. And this tells us about disparity in medicine. In fact, we didn't see these effects in health controls. In chronic pain patient, we also saw a sex concordance influence, but in the opposite sense. In women studied by a man experimenter, placebo effects are larger. Such an effect was not seen in men. The other question that we had was, what about the contribution of psychological factors? At that stage, there were many different surveys used by different labs, somehow based on a different area of, the states, the world. There were trends where in some people, in some study, they observe an effects of neuroticism, positive and negative asset, bis, bus, that refer to rewards. Instead of focusing on a single survey, and now we have a beautiful meta-analysis say that is not worthy in the sense that it's not predictive of placebo effects. We use the ROC model suggested by the NIMH, and by doing a sophisticated approach, we were able to combine this into 4 valence: emotional distress, reward seeking, pain-related fear and pain catastrophizing, empathy, and openness. These 4 valence then were interrogated to predict placebo effects. And you can see that emotional distress is associated with lower magnitude of placebo effects, extinguishing over time, and lower proportion of placebo responsivity. Also, people who tend to catastrophizing display lower magnitude of placebo effects. In terms of expectation, it's also interesting. Patients expect to benefit. They have this desire for a reward, but also those people who are more open and characterized by empathy tend to have larger expectations, but this doesn't translate necessarily in larger placebo effects, somehow hinting that the two phenomena can be not necessarily linked. Because we study chronic pain patients, they come with their own baggage of disease, comorbidities, and Dr. Wang in the department look at insomnia. Those people suffering from insomnia tend to have lower placebo analgesic effects, along with those who have a poor pattern of sleep, suggesting that clinical factor can be relevant when we wish to predict placebo effects. Another question that we address how simple SNPs, single nucleotide polymorphism variants in 3 genes that have been published can be predictive of placebo effects. In particular, I'm referring to OPRM1 that is linked to the gene for endogenous opioids, COMT linked to endogenous dopamine, and FA linked to endocannabinoids. And we will learn more about that with the next talk. And you can see that there is a prediction, these are rock curves, that can be interesting. We model all participants, verbal suggestion alone, conditioning. There is really a huge difference between using 1 SNP versus 2 or 3. What truly impacted and was stronger in terms of prediction was accounting for the procedure we use to study placebo, verbal suggestion alone versus conditioning. When we add the manipulation, the prediction becomes stronger. More recently, we studied gene expression, transcriptomic profile associated with placebo effects. We select from the 402 participants randomly 50/40 MD, and we extract their transcriptomic profiles. Also, we select a validation cohort to see if we can replicate what we discover in terms of RNA sequencing. So we found over 600 genes associated with the discovered cohort. In blue are the genes downregulated and in red upregulated. We chose the top 20 genes and we did the PCA to validate the top 20, and we found that 6 of them were replicated, and they include all these genes that you see here. This element for us was particularly interesting, as well as the PI3, the CDC85B, XBXL15, H, AGHL, and the TNF RSF4. So with this...
Hi, Doctor, you have 1 minute remaining.
Yeah, I'm done. With this, that is the goal probably one day with AI and other approach to combine clinical, psychological, brain imaging, and so on characteristic and be able to predict the level of trend to respond to placebo. That may guide us in clinical trials and clinical practice, well, tailor the treatments. Therefore, placebo and nocebo biological response can be to some extent predicted, and identify those who respond to placebo can help tailoring drug development and symptom management. Thank you to my lab, to all of you, the funding agencies, and finally, for those who like to read more about placebo, this book is available for free to be downloaded, and include many of the speakers from this 2-day event as contributors to this book. Thank you very much.
Thank you so much, Luana. It was a wonderful presentation. We have one question in the Q&A. And is the elegant studies demonstrating powerful phenomena? 2 questions. Is it possible to extend or sustain placebo boosting effect? And what's the dose response relationship with placebo or nocebo?
Great questions. The goal is to boost placebo effects. And one way, as I showed, was, for example, using intranasal vasopressin. For the dose extending relationship with placebo, we know that we need a minimum of 3-4 administration before boosting this sort of pharmacological memory. And the longer is the administration of the active drug before we replace with placebo, the larger the magnitude of placebo effects. For nocebo, we show similar relationship together with Thor and other collaborators. So again, the longer we condition, the stronger the placebo or nocebo effect. Thank you so much.
Maybe I wanted to ask, do you have any theory and interpretation about the potential for transmit to pass on a placebo response between the observer, more social? Do you have any interpretation of this phenomenon?
It is not completely new in the literature. There is a lot of studies show that we can transfer pain in both animal models and humans. So transfer analgesia is a natural continuation of that line of research. And the fact that we mimic things that we see in some other people, this is the very most basic form of learning when we grow up, but also from an evolutionary point of view, protect us from predators when animals and us as humans begin. So observing is a very good mechanism to boost behaviors, and in this case, placebo effects. Thank you.
Okay. We will have more time to answer questions. We are going to move on to the next speaker, Dr. Catherine Hall.
Thank you. Can you see my screen okay? Great. So I'm going to build on Dr. Koloka's talk to really kind of give us a deeper dive into the genetics of the placebo response in clinical trials. So I have no disclosures. So as we heard, and as we've been hearing over the last 2 days, there are physiological drivers of placebo effects, whether they're opioid signaling or dopamine signaling. And these are potentiated by the administration, or can be potentiated by saline injections, sugar pills. And what's really interesting here, I think, is this discussion about how drugs impact the drivers of placebo response. In particular, we heard about naloxone yesterday and proglumide. And what I want to do really today is to think about the next layer, like how do the genes that shape our biology and really drive or influence those physiological drivers of placebo response, how do the genes modify our placebo response, but also how are they modifying the effect of the drugs and the placebos on this network? And if you think about it, we really don't know much about all the many interactions that are happening here. And I would actually argue that it goes even beyond genetic variation to other factors that lead to heterogeneity in clinical trials. But today I'm going to really focus on genes and variations in the genome. So let's go back so we have the same terminology. I'm going to be talking about placebo response in trials. And so we saw this graph, or a version of this graph yesterday, where in clinical trials, when we want to assess the effect of a drug, we subtract the outcomes in the placebo arm from the outcomes in the drug treatment arm. And there's a basic assumption here that the placebo response is additive to the drug response. And what I want to do today is to really challenge that assumption. I want to challenge that expectation, because I think we have enough literature and enough studies that have already been done that demonstrate that things are not as simple as that, and that we might be missing a lot from this basic averaging and subtracting that we're doing. So the placebo response is the stippled lines there, which includes placebo effects, which we've been focusing on here, but it also includes a natural history of the disease or the condition, phenomena such as statistical regression to the mean, blinding and bias, and Hawthorne effects. So we lump all those together in the placebo arm of the trial and subtract the placebo response from the drug response to really understand the drug effect. So one way to ask about, well, how do genes affect this is to look at candidate genes. And as Dr. Koloka pointed out, and has done some very elegant studies in this area, genes like COMT, opioid receptors, genes like OPRM1, the Fa endocannabinoid signaling genes are all candidate genes that we can look at in clinical trials and ask, did these genes modify what we see in the placebo arm of trials? We did some studies in COMT, and I want to just show you those to get so you can get a sense of how genes can influence placebo outcomes. So COMT is catecholomethyltransferase, and it's a protein, an enzyme that metabolizes dopamine, which, as you saw, is important in mediating the placebo response. COMT also metabolizes epinephrine, norepinephrine, and catechol-estrogen. So the fact that COMT might be involved in placebo response is really interesting because it might be doing more than just metabolizing dopamine. So we asked the question, what happens if we look at COMT genetic variation in clinical trials of irritable bowel syndrome? And working with Ted Kapchuk and Tony Lambeau at Beth Israel Deaconess Medical Center, we did just that. We looked at COMT effects in a randomized clinical trial of irritable bowel syndrome. And what we did see was that for the gene polymorphism RS4680, we saw that people who had the weak version of the COMT enzyme actually had more placebo response. These are the met people here shown on this by this arrow, and that the people who had less dopamine, because that enzyme didn't work as well for this polymorphism, they had less of a placebo response in one of the treatment arms. And we would later replicate this study in another clinical trial that was recently concluded in 2021. So to get a sense, as you can see, we started off being somewhat limited by what was available in the literature. And so we wanted to expand on that to say more about genes that might be associated with placebo response. So we went back and we found 48 studies in the literature where there was a gene that was looked at that modified the placebo response. And when we map those to the interactome, which is this constellation of all gene products and their interactions, their physical interactions, we saw that the placebo or the placebo module had certain very interesting characteristics. Two of those characteristics that I think are relevant here today are that they overlapped with the targets of drugs, whether they were analgesics, antidepressive drugs, anti-Parkinson's agents. Placebo genes putatively overlapped with drug treatment genes or targets. They also overlapped with disease-related genes. And so what that suggests is that when we're looking at the outcomes of a clinical trial, there might be a lot more going on that we're missing. And let's just think about that for a minute. On the left is what we expect. We expect that we're going to see an effect in the drug. It's going to be greater than the effect of the placebo, and that difference is what we want, that drug effect. But what we often see is on the right here, where there's really no difference between drug and placebo. And so we're left to scratch our heads. Many companies go out of business. Many sections of companies close. And quite frankly, patients are left in need. Money is left on the table because we can't discern between drug and placebo. And I think what's interesting is that's been a theme that's kind of arisen since yesterday, where, oh, if only we had better physiological markers or better genes that targeted physiology, then maybe we could see a difference and we can, move forward with our clinical trials. But what I'm going to argue today is actually what we need to do is to think about what is happening in the placebo arm, what's contributing to the heterogeneity in the placebo arm. And I'm going to argue that when we start to look at that compared to what's happening in the drug treatment arm, oftentimes, and I'm going to give you demonstration after demonstration, and believe me, this is just the tip of the iceberg, what we're seeing is that there are differential effects by genotype in the drug treatment arm and the placebo treatment arm, such that if you average out what's happening in these drug and placebo arms, you would basically see that there's no difference. But actually, there's some people that are benefiting from the drug, but not placebo, and conversely, benefiting from placebo, but not drug. Averages out to no difference. Let me give you some examples. We had this hypothesis, and we started to look around to see if we could get partners who had already done clinical trials that had happened to have genotyped COMT. And what we saw in this clinical trial for chronic fatigue syndrome, where adolescents were treated with clonidine, was that when we looked in the placebo arm, we saw that the VAL-VAL patients, so this is the COMT genotype, this is the low activity COMT genotype, sorry, that's high activity COMT genotype. They had the largest increase in the number of steps they were taking per week. In contrast, the MET people, the people with the weaker dopamine COMT, had fewer, almost no change in the number of steps they were taking per week. So you would look at this and you would say, oh, the VAL-VAL people were the placebo responders and the MET people didn't respond to placebo. But what we saw when we looked in the drug treatment arm was very surprising. We saw that clonidine literally erased the effect that we were seeing in placebo for the VAL-VAL participants in this trial. And clonidine basically was having no effect on the heterozygotes, the VAL-METs, or on the MET-METs. And so this trial rightly concluded that there was no benefit for clonidine, but if they hadn't taken this deeper look at what was happening, they would have missed that clonidine may potentially be harmful for people with chronic fatigue in this particular situation. And so what we really need to do, I think, is to look not just in the placebo or not just in the drug treatment arm, but in both arms to understand what's happening there. And I'm going to give you another example. And like I said, the literature is replete with these examples. On the left is an example from a drug that was used to test cognitive skills, tolcapone, which actually targets COMT. And what you can see here, again, on the left, is differential outcomes in the placebo arm and in the drug treatment arm that if you were to just average these two, you would not see the differences. On the right is a really interesting study looking at alcohol among people with alcohol disorder, a number of % drinking days. And they looked at both COMT and OPRM1. And this is what Dr. Koloka was just talking about, that there seemed to be not just gene placebo drug interactions, but gene drug placebo interactions. This is a complicated space. And I know we like things to be very simple, but I think what these data are showing us is we need to pay more attention. So let me give you another example, because these, you could argue, okay, those are objective outcomes, subjective outcomes. Let's take a look at the Women's Health Study, arguably one of the largest studies on aspirin versus placebo in history. 30,000 women were randomized to aspirin or placebo, and lo and behold, after 10 years of following them, the p-value was non-significant. There was no difference between drug and placebo. So we went to this team and we asked them if could we look at COMT, because we had a hypothesis that COMT might modify the outcomes in the placebo arm and potentially differentially modify the outcomes in the drug treatment arm. And you might be saying, well, that can't have anything to do with a placebo effect. And we completely agree. This, if we did find it, would suggest that there might be something to do with the placebo response that's related to natural history. And I'm going to show you the data what we found. So when we compared the outcomes in the placebo arm to the aspirin arm, what we found was the MET women randomized to placebo had the highest of everybody rates of major cardiovascular disease, which means the highest rates of myocardial infarction, stroke, revascularization, and death from a cardiovascular disease cause. In contrast, the MET women on aspirin had benefit, had a statistically significant reduction in these rates. Conversely, the VAL-VAL women on placebo did the best, but the VAL-VAL women on aspirin had the highest rates, had significantly higher rates than the VAL-VAL women on placebo. What does this tell us? Well, we can't argue that this is a placebo effect because we don't have the control for placebo effects, which is a no treatment control. But we can say that these are striking differences that, like I said before, if you don't pay attention to them, you miss the point, that there's subpopulations for benefit or harm because of differential outcomes in the drug and placebo arms of the trial. And so I'm going to keep going. There are other examples of this. We also partnered with a group at Brigham and Women's Hospital that had done the CAMP study, the Childhood Asthma Management Study. And in this study, they randomized patients to placebo, budesonide, or nedocromil for 5 years and studied asthma outcomes. Now, what I was showing you previously was candidate gene analyses. What this was a GWAS. We wanted to be agnostic and ask, are there genes that modify the placebo outcomes, and are these outcomes different when we look in the drug treatment arm? And so in that little inset is a picture of all the genes that were looked at in a GWAS, and we had a borderline genome-wide significant hit called BBS9. And when we looked at BBS9 in the placebo arm, those white boxes at the top are basically the baseline levels of coughing and wheezing among these children. And in the gray are at the end of the treatment, their level of coughing and wheezing. And what you can see here is that participants with the AA genotype were the ones that benefited from budesonide, from placebo, whereas the patients with the GG genotype, really, there was no significant change. Now, when we looked in the drug treatment arms, we were surprised to see that the outcomes were the same, of course, at baseline. Everybody's kind of the same, but you can see their differential responses depending on your genotype. And so again, not paying attention to these gene, drug / placebo interactions, we miss another story that is happening here among our patients. Now, I added this one because it's important to realize that this is not just about gene, drug, placebo, but these are also about epigenetic effects. And so here's the same study that I showed earlier on alcohol use disorder. They didn't just stop at looking at the polymorphisms or the genetic variants. This team also went so far as to look at methylation of OPRM1 and COMT. So methylation is basically when the promoter region of a gene is basically blocked because it has a methyl group. It has methylation on some of the nucleotides in that region. So you can't make the protein as efficiently. And if you look on the right here, what you can see in these 3 models that they looked at other genes. They also looked at SLC6A3 that's involved in dopamine transport. And what you can really see here is that there are significant gene by group by time interactions for all these 3 genes. These are candidate genes that they looked at. And even more fascinating is their gene by gene interactions. Basically, it's saying that you cannot say what the outcome is going to be unless you know the patient's or the participants' COMT or OPRM genotype A, and also how methylated the promoter region of these genes are. So this makes for a very complicated story, and I know we like very simple stories, but I want to say that I'm just adding to that picture that we had before to say that it's not just in terms of the genes, polymorphisms, but as Dr. Koloka just elegantly showed, it's transcription as well as methylation that might be modifying what's happening in the drug treatment arm and the placebo treatment arm. And to add to this, it might also be about the natural history of the condition. So BBS9 is actually a gene that's involved in the cilia, the activity of the formation of the cilia, which is really important in breathing in the nasal canal. And so you can see that it's not just about what's happening in the moment when you're doing the placebo or drug or the clinical trial. It also the genes might also be modifying where the patient starts out and how the patient might develop over time. So in essence, we have a very complicated playground here, but I think I've shown you that genetic variation, whether it's polymorphisms in the gene-gene interactions, or epigenetics, or all of the above, can modify the outcomes in placebo arms of clinical trials. And that this might be due to the genetic effects on placebo effects or the genetic effects on natural history. And this is something I think we need to understand and really pay attention to. And I also think I've showed you, and these are just a few examples, there are many more, that genetic variation can differentially modify drugs and placebos, and that these potential interaction effects really challenge this basic assumption of additivity that I would argue we've had for far too long, and we really need to rethink.
Very cool.
Hi, Ted.
Oh, I didn't know I was on. That was
Yeah,
so cool.
That's great. That's great. So in summary, can we use these gene drug placebo interactions to improve clinical trials? Can we change our expectations about what's happening? And perhaps, as we've been saying for the last 2 days, we don't need new drugs with clear physiological effects. What we need is to understand drug and placebo interactions and how they impact subpopulations and can reveal who benefits or is harmed by therapies. And finally, as we started to talk about in the last talk, can we use drugs to boost placebo responses? Perhaps some drugs already do. Conversely, can we use drugs to block placebo responses? And perhaps some drugs already do. So I just want to thank my collaborators. There was Ted Kapchuk, one of my very close and dear mentors and collaborators, and really thank you for your time.
Thank you so much. It was a terrific presentation, and definitely Ted Kapchuk liked it. It was just spontaneous, the best spontaneous laughs. We have a couple of questions coming through the chat. One is about the heterogeneity of response in placebo arms. It's not uncommon to see quite a dispersion of response in such trials. Propose a thought experiment. If one look at the fraction of high responders in the placebo arms, would one expect it to see enrich for some of the genetic markers for enhanced placebo response?
I absolutely think so. We haven't done that. And I would argue that, you know, we've been having a kind of quiet conversation here about naloxone, because I think Lauren yesterday said that the findings of naloxone are variable, that sometimes it looks like naloxone is blocking placebo response and sometimes it isn't. And I would argue that we need to know more about who is in that trial, right? Is this that, because I could have gone on and showed you that there's differences by gender, right? And so this heterogeneity that's coming into clinical trials is not just coming from the genetics, it's coming from race, ethnicity, gender, population. Like, are you in Russia, or are you in China, or are you in the US when you're doing your clinical trial? We really need to start unpacking this and paying attention to it, because I think because we're not paying attention to it, we are wasting a lot of money.
And epigenetic is another way to consider traumatic experiences, adverse event, learning. There's another component that we're not tracking accurately in clinical trials. I don't think this is one of the elements routinely collected, especially in antidepressant clinical trials, is just now coming to the surface. Thank you. Another question comes, it says the different approaches. One is GWAS versus candidate gene approach. How do you start to think about genes that have a potential implication in neurophysiological pathways and choosing candidate to test versus a more agnostic GWAS approach?
I believe you have to do both. And because you don't know what you're going to find if you do a GWAS, and it's important to know what's there. At the same time, I think it's also good to test our assumptions and to replicate our findings, right? And so once you do the GWAS and you have a finding, for instance, our BBS9 finding would be amazing to replicate or to try and test in another cohort. But of course, it's really difficult to do a whole clinical trial again. These are very expensive and they last many years. And so, you know, I think replication is something that's tough to do in this space, but is really important. And I would do both.
Thank you. I think we're a little short on time. We are going to move on to the next speaker. Thank you so much.
Good morning. It's me, I imagine.
Yes, that's it.
Or good afternoon. Let me share my screen. Yeah, so good morning. This is going to be a tough act to follow. Dr. Coloca and Dr. Hall's presentations were really elegant, so manage your expectations for mine. And Ted, please feel free to unmute yourself, because I think your laugh is incredibly contagious, and I think we all were laughing as well. So my name is Fadel Zidan. I'm at UC San Diego, and I'll be discussing mostly unpublished data that we have that's under review, examining if and how mindfulness meditation assuages pain, and if the mechanisms supporting mindfulness meditation-based analgesia are distinct from placebo. And so, this is kind of like a household slide that we all are here because we all appreciate how much of an epidemic chronic pain is, and how significant it is, how much it impacts our society and the world. And it is considered a silent epidemic because of the catastrophic and staggering costs to our society. And that's largely due to the fact that the subjective experience of pain is modulated and constructed by a constellation of interactions between sensory, cognitive, emotional dimensions, genetics. The list can go on. And so what we've been really focused on for the last 20 years or so is to appreciate if there is a non-pharmacological approach, a self-regulated approach that could be used to directly assuage the experience of pain, to acutely modify exacerbated pain. And to that extent, we've been studying meditation, mindfulness-based meditation. And mindfulness is a very nebulous construct. If you go from one lab to another lab to another lab, you're going to get a different definition of what it is. But obviously, my lab's definition is the correct one. And so the way that we define it is awareness of arising sensory events without reaction, without judgment. And we could develop this construct, this disposition, by practicing mindfulness-based meditation, which I'll talk about here in a minute. And we've seen a lot of, and this is an old slide, a lot of new evidence, converging evidence, demonstrating that 8 weeks of manualized mindfulness-based interventions can produce pretty robust improvements in chronic pain and opiate misuse. These are mindfulness-based stress reduction programs, mindfulness-oriented recovery enhancement, mindfulness-based cognitive therapy, which are about 8 weeks long, 2 hours of formalized didactics a week, 45 minutes a day of homework. There's yoga, there's mental imagery, breathing meditation, walking meditation, a silent retreat, and about a $600 tab, which may not be, although they're incredibly effective, may not be targeting demographics and folks that may not have the time and resources to participate in such an intense program. And to that extent, as an immigrant to this country, I've noticed that we kind of are like this drive-through society where, we have a tendency to eat our lunches and our dinners in our cars. We're attracted to really brief interventions for exercise or anything really, pharmaceuticals, like 8 minutes of abs and buns of steel. And we even have things called like the military diet that promise that you would lose 10 pounds in 3 days without dying. So we seemingly are attracted to these fast-acting interventions. And so to this extent, we've worked for quite some time to develop a very user-friendly, very brief mindfulness-based intervention. So this is an intervention that's about 4 sessions, 20 minutes each session. And participants we remove all religious aspects, all spiritual aspects, and we really don't even call it meditation. We call it mindfulness-based mental training. And our participants are taught to sit in a straight posture, close their eyes, and to focus on the changing sensations of the breath as they arise. And what we've seen is this repetitive practice enhances cognitive flexibility and the ability to sustain attention. And when individuals' minds drift away from focusing on the breath, they're taught to acknowledge distracting thoughts, feelings, emotions without judging themselves or the experience, doing so by returning their attention back to the breath. So there's really a 1-2 punch here where A, you're focusing on the breath and you're enhancing cognitive flexibility, and B, you're training yourself to not judge discursive events, and that we believe enhances emotion regulation. So quite malleable to physical training, we would say mental training. Now that we have the advent of imaging, we could actually see that there are changes in the brain related to this. But as many of you know, mindfulness is kind of like a household term now. It's all over our mainstream media. We have, LeBron meditating courtside, Oprah meditating with her Oprah blanket, Anderson Cooper's meditating on TV, and Time Magazine puts, people on the cover meditating, and it's just all over the place. And so these types of images and these types of, I guess, insinuations could elicit non-specific effects related to meditation. And for quite some time, I've been trying to really appreciate, not is meditation more effective than placebo, although that's interesting, but does mindfulness meditation engage mechanisms that also are shared by placebo? So beliefs that you're meditating could elicit analgesic responses. The majority of the manualized interventions, in their manuals, they use terms like the power of meditation, which I guarantee you is analgesic. To focus on the breath, we need to slow the breath down. Not explicitly, but it just happens naturally. And slow breathing can also reduce pain, facilitate attention, social support, conditioning, all factors that are shared with other therapies and interventions, but in particular are also part of meditation training. So the question is, because of all this, is mindfulness meditation merely, or not merely, after these 2 rich days of dialogue, but is mindfulness meditation engaging processes that are also shared by placebo? So if I apply a placebo cream to someone's calf and then throw them in the scanner versus asking someone to meditate, the chances are very high that the brain processes are going to be distinct. So we wanted to create and validate an operationally matched mindfulness meditation intervention that we coined as sham mindfulness meditation. It's not sham meditation because it is meditation. It's a type of meditative practice called pranayama. But here in this intervention, we randomize folks, we tell folks that they've been randomized to a genuine mindfulness meditation intervention, straight posture, eyes closed, and every 2 to 3 minutes, they're instructed to, quote unquote, take a deep breath as we sit here in mindfulness meditation. We even match the time giving instructions between the genuine and the sham mindfulness meditation intervention. So the only difference between the sham mindfulness and the genuine mindfulness is that the genuine mindfulness is taught to explicitly focus on the changing sensations of the breath without judgment. The sham mindfulness group is just taking repetitive, deep, slow breaths. So if the magic part of mindfulness, if the active component of mindfulness is this non-judgmental awareness, then we should be able to see disparate mechanisms between these. And then we also use a third arm, a book listening control group called The Natural History of Cell Burn, where it's a very boring, arguably emotionally pain-evocating book for 4 days. And this is meant to control for facilitator time and, facilitator attention and the time elapsed in the other group's interventions. So we use a very high level of noxious heat to the back of the calf. And we do so because imaging is quite expensive, and we want to ensure that we could see pain-related processing within the brain. Here, across all of our studies, we use 10, 12-second plateaus of 49 degrees to the calf, which is pretty painful. And then we assess pain intensity and pain unpleasantness using a visual analog scale, where here the participants just see like red. The more they pull on the algometer, the more in pain they are. But on the back are numbers for us, where 0 is no pain and 10 is the worst pain imaginable. And so pain intensity can be considered like sensory dimension of pain, and pain unpleasantness could be more of like, I don't want to say pain affect, but more like the bothersome component of pain unpleasantness. So what we did was we combined all of our studies that have used the mindfulness, sham mindfulness, and this book listing control to see, does mindfulness meditation engage is mindfulness meditation more effective than sham mindfulness meditation at reducing pain? And we also combine 2 different fMRI techniques: blood oxygen dependent level signaling, BOLD, which allows us a higher temporal resolution and signal to noise ratio than, let's say, a perfusion imaging technique, and allows us to look at connectivity. Our meditation is also predicated on changes in respiration rate, which could elicit pretty dramatic artifacts in the brain, breathing-related artifacts explicitly related to CO2 output. So using a perfusion-based fMRI technique like arterial spin labeling is really advantageous as well, although it's not as temporally resolute as BOLD. It provides us a direct quantifiable measurement of cerebral blood flow. So straight to the results. On the y-axis, we have the change in pain ratings. And on the x-axis are book listening controls, sham mindfulness meditation, mindfulness meditation. Here are our large sample sizes. Blue is intensity and red is unpleasantness. And this is the post-intervention fMRI scans where we see that from the first half of the scan to the second half of the scan, our control participants are simply resting, and pain just increases because of pain sensitization and being in a claustrophobic MRI environment. And you can see here that sham mindfulness meditation does produce pretty significant reductions in pain intensity and unpleasantness, more than the control group. But mindfulness meditation is more effective than sham mindfulness and the controls at reducing pain intensity and pain unpleasantness. So there does seem to be some kind of additive component to the genuine intervention, although this is a really easy practice, the sham technique. So, you know, for folks that have maybe fatigue or cognitive deficits or just, you know, aren't into doing the mindfulness technique, I highly recommend this technique, which is just a slow breathing approach. And it's dead easy to do. Anyone that's practiced mindfulness for the first time or a few times can state that it can be quite difficult and what's the word? Involving, right? Okay, so what happened in the brain? So these are our CBF maps from 2 studies that we replicated in 2011 and 15, where we found that higher activity, higher CBF in the right anterior insula, which is ipsilateral to the stimulation site, and higher rostral anterior cingulate cortex subgenual ACC was associated with greater pain relief, pain intensity. And in the context of pain unpleasantness, higher orbitofrontal cortical activity was associated with lower pain. And this is very reproducible, where we see greater thalamic deactivation predicts greater analgesia on the unpleasantness side. These areas, obviously right anterior insula in conjunction with other areas, is associated with interoceptive processing, awareness of somatic sensations. And then the ACC and the OFC are associated with like higher order cognitive flexibility, emotion regulation processes. And the thalamus is really the gatekeeper from the brain, from the body to the brain. Nothing can enter the brain except for, unless it goes through the thalamus, except if it's the sense of smell. So it's really like this gatekeeper of arising nociceptive information. So to take home here is that mindfulness is engaging multiple neural processes to assuage pain. It's not just one singular pathway. Our BOLD studies were also pretty insightful. Here we ran a PPI analyses, psychophysiological interaction analyses, and this was whole brain to see what brain regions are associated with pain relief in the context of using the BOLD technique. And we find that greater ventral medial prefrontal cortical activity deactivation, is associated with lower pain. And the VMPFC is a super evolved area that's associated with like higher order processes relating to self. It's the central node, one of the central nodes of the so-called default mode network, a network supporting self-referential processing. But in the context of the VMPFC, I like the way that Tor and Matu reflect the VMPFC as being more, related to affective meaning. And there's been really nice papers showing that VMPFC is uniquely involved in quote unquote self-ownership or subjective value, which is particularly interesting for the context of pain, because pain is a very personal experience that's directly related to the interpretation of arising sensations and what they mean to us. And seemingly, and I apologize for the reverse inferencing here, but seemingly mindfulness meditation based on our qualitative assessments as well is reducing the ownership or what the intrinsic value, the contextual value of those painful sensations, i.e. they don't feel like they bother, that pain is there, but it doesn't bother our participants as much, which is quite interesting as a manipulation. We also ran a connectivity analysis between the contralateral thalamus and the whole brain. And we found that greater decoupling between the contralateral thalamus and the precuneus, another central node of the default mode network, predicted greater analgesia. So this is a really cool, I think, together mechanism showing that two separate analyses are indicating that the default mode network could be an analgesic system, which we haven't seen before. We have seen the DMN involved in chronic pain and pain-related exacerbations, but I don't think we've seen it as being a part of an analgesic, like being a pain-relieving mechanism. Interestingly, the thalamus and the precuneus together are the first two nodes to go offline when we lose consciousness, and they're the first two nodes to come back online when we recover from consciousness, suggesting that that the thalamus and the precuneus are involved in self-referential awareness, consciousness of self, things of this nature. So again, multiple processes involved in meditation-based pain relief, which maybe gives rise to why we are seeing consistently that meditation could elicit long-lasting improvements in pain unpleasantness in particular, as compared to sensory pain, although it does that as well. And also, the data gods were quite kind on this because, these mechanisms are also quite consistent with the primary premises of Buddhist and contemplative scriptures, saying that the primary principle is that your experiences are not you. You are there, not that there is no self, but that the processes that arise in our moment-to-moment experience are merely reflections and interpretations and judgments. And that may not be the true inherent nature of mind. And so before I get into more philosophical discourse, I'm going to keep going for the sake of time. Okay, so what happened with the sham mindfulness meditation intervention? Well, we did not find that any neural processes predicted analgesia significantly during sham mindfulness meditation. What did predict analgesia during sham mindfulness was slower breathing rate, which we have never seen before with mindfulness. We've never seen a significant or even close to significant relationship between mindfulness-based meditation, analgesia, and slow breathing. But over and over and over, we see that sham mindfulness-based analgesia is related to slower breathing, which provides us this really cool, distinct process where kind of this perspective where mindfulness is engaging higher order, top-down type processes to assuage pain, while sham mindfulness may be engaging this more bottom-up type response to assuage pain. And so I'm going to move on to some other new work. And this is in a great collaboration with the lovely Tor Wager, and he's developed these with Marta and Wu, these wonderful signatures, these machine learned multivariate pattern signatures that are remarkably accurate at predicting pain, over I think like 98, 99%. His seminal paper, The Neurological Pain Signature, was published in New England Journal of Medicine, that showed that these signatures can predict nociceptive specific, in particular for this paper, thermal heat pain, with incredible accuracy. And it's not modulated by placebo or affective components per se. And then the SIPS is a machine learned signature that is, as they put it, associated with cerebral contributions to pain. But if you look at it closely, it's really, these are markers that are highly responsive to the placebo response. So the SIPS can be used, and he has this beautiful preprint out showing that it does respond with incredible accuracy to placebo, varieties of placebo. So we use this MVPA to see if meditation engages signatures supporting placebo responses. And then Marta Checo's latest paper with Tor, published in Nature Neuro, found that the negative affective signature predicts pain responses above and beyond nociceptive related processes. So this is pain related to negative affect, which again contributes to the multimodal processing of pain, and how now we could use these elegant signatures to kind of disentangle which components of pain meditation and other techniques assuage. Here's the design. We had 40, we combined two studies, one with BOLD and one with ASL. So this will be the first ASL study with signatures, with these MVPA signatures. And we had the mindfulness interventions that I described before, the book listing interventions I described before, and a placebo cream intervention, which I'll describe now, all in response to 49 degrees thermal stimuli. So across, again, all of our studies, we use the same methods. And the placebo group, and I'll try to be quick about this, this is a kind of a combination of Luana Coloca, Don Price, and Tor's placebo conditioning interventions, where we administer 49 degrees. Well, we tell our participants that we're testing a new form of lidocaine. And the reason that it's new is that the more applications of this cream, the stronger the analgesia. So in the conditioning sessions, they come in, administer 49 degrees, apply and remove this cream, which is just petroleum jelly, after 10 minutes. And then we covertly reduce the temperature to 48. And then they come back in session 2 and 3. After 49 degrees and removing the cream, we lower the temperature to 47. And then on the last conditioning session, after we remove the cream, we lower the temperature to 46.5, which is a qualitatively completely different experience than 49. And we do this to lead our participants to believe that the cream is actually working. And then in a post-intervention MRI session, after we remove the cream, we don't modulate the temperature. We just keep it at 49, and that's how we measured placebo in these studies. And so here, again, is John Dean and Gabe are co-leading this project. Here, pain intensity on this axis, pain unpleasantness on that axis. Controls from the beginning of the scan to the end of the scan significantly go up in pain. Placebo cream was effective at reducing intensity and unpleasantness, but we see mindfulness meditation was more effective than all the conditions at reducing pain. The signatures, we see that the nociceptive specific signature, the controls go up in pain here. No change in the placebo, and then mindfulness meditation, you can see here, produces a pretty dramatic reduction in the nociceptive specific signature. The same is true for the negative affective signature. Mindfulness meditation uniquely modifies this signature as well, which I believe this is one of the first studies to show something like this. But it does not modulate the placebo signature. What does modulate the placebo signature is our placebo cream, which is a really nice manipulation check for these signatures. So here, taken together, we show that mindfulness meditation, again, is engaging multiple processes and is reducing pain by directly assuaging nociceptive specific markers, as well as markers supporting negative affect, but not modulating placebo-related signatures, providing further credence that it's not a placebo-type response. And we're also demonstrating this granularity between a placebo mechanism that's not being shared by another active mechanism. While we all assume that active therapies and techniques are using a shared subset of mechanisms or processes with placebo, here we're providing accruing evidence that mindfulness is separate from a placebo. And I'll try to be very quick on this last part. This is all not technically related to placebo, but I would love to hear everyone's thoughts on these new data we have. So as we've seen elegantly, that pain relief by placebo, distraction, acupuncture, transcranial magnetic stimulation, prayer are largely driven by endogenous opioidergic release. Yes, there are other systems. Other system is the endocannabinoid system, serotonergic system, dopamine. The list can go on, but it's considered by most of us that the endogenous opioidergic system is the central pain modulatory system. And the way we do this is by antagonizing endogenous opioids by employing incredibly high administration dosage of naloxone. And I think this wonderful paper by Siri Leknes's group provides a nice primer on the appropriate dosages for naloxone to antagonize opiates. And I think a lot of the discussions here where we see differences in naloxone responses are really actually reflective of differences in dosages of naloxone. It metabolizes so quickly that I would highly recommend a super large bolus with a maintenance infusion IV. And we've seen this to be a quite effective way to block endogenous opioids. And across 4 studies now, we've seen that mindfulness-based pain relief is not mediated by endogenous opioids. It's something else. We don't know what that something else is, but we don't think it's endogenous opioids. But what if it's sex differences that could be driving these opioidergic versus non-opioidergic processes? We've seen that females exhibit higher rates of chronic pain than males. They are prescribed opiates at a higher rates than men. And when you control for weight, they require higher dosages than men. Why? Well, there's extant literature in rodent models and preclinical models that demonstrate that male rodents versus male rodents engage endogenous opioids to reduce pain, but female rodents do not. And this is a wonderful study by Anne Murphy that basically shows that males in response to morphine have a greater latency in Paul withdrawal coupled with morphine, and not so much with females. But when you add naloxone to the picture with morphine, the latency goes down. It basically blocks the analgesia in male rodents, but it enhances analgesia in female rodents. So we basically asked, Michaela, an undergraduate student doing an honors thesis, asked this question: well, are males and females in humans engaging distinct systems to assuage pain? So she really took off with this. And here's the design. We had noxious heat in the baseline.
Doctor, you have 1 minute left. Can you wrap up?
Yep. Let me just, basically, we asked, are there sex differences between males and females during meditation in response to noxious heat? And there are. Baseline, just change in pain. Green is saline, red is naloxone. You could see that with naloxone on board, there's greater analgesia in females, and we reversed the analgesia largely. There's no differences between baseline and naloxone in males, and the males are reducing pain during saline. So we believe this is the first study to show something like this in humans. Super exciting. It also blocked the stress reduction response in males, but not so much in females. Let me just acknowledge our funders, some of our team, and I apologize for the fast presentation. Thank you.
Thank you so much. That was awesome. We are a little bit short in time. I would suggest to go into a 10-minute break. We're going to reconvene at 1:40. And please continue to add your questions to the Q&A, and our speakers are going to answer, or we're going to bring some of those questions directly to the discussion panel at the end of the session today. Thank you so much. Hello, welcome back. I'm really honored to introduce our next speaker, Dr. Marta Pesinha, and she's going to talk about mapping expectancy mood interactions in antidepressant placebo effects. Thank you so much.
Thank you, Christina. It's my great pleasure to be here and just I'm going to switch gears a little bit to talk about antidepressant placebo effects. And in particular, I'm going to talk about the relationship between acute expectancy mood neurodynamics and long-term antidepressant placebo effects. So, well, we all know that depression is a very prevalent disorder. And yes, in 2020, major depressive disorder affected 21 million adults in the US and 280 million adults worldwide. And current projections indicate that by the year 2030, it will be the leading cause of disease burden globally. Now, response rates to first-line treatments, antidepressant treatments, are approximately 50%, and complete remission is only achieved in 30 to 35% of individuals. Also, depression tends to be a chronic disorder, with 50% of those recovering from a first episode having an additional episode, and 80% of those with 2 or more episodes having another recurrence. And so for patients who are non-responsive to 2 intervention, remission rates with subsequent therapy drop significantly to 10 to 25%. And so in summary, we're facing a disorder that is very resistant or becomes resistant very easily. And in this context, one would expect that antidepressant placebo effects would actually be low. But we all know that this is not the case. The response rate to placebos is approximately 40% compared to 50% response rates to antidepressants. And obviously, this varies across the studies. But what we do know and learned yesterday as well is that response rates to placebos have increased approximately 7% over the last 40 years. And so this high prevalence of placebo response in depression have significantly contributed to the current psychopharmacology crisis, where large pharma companies have reduced at least in half the number of clinical trials devoted to CNS disorders. Now, antidepressant placebo response rates among individuals with depressions are higher than in any other psychiatric condition. And this was recently published again in this meta-analysis of approximately 10,000 psychiatric patients. Now, other disorders where placebo response rates are also prevalent are generalized anxiety disorder, panic disorders, HDHD, or PTSD, and maybe less frequent, although still there, in schizophrenia or OCD. Now, importantly, placebo effects occur not only in response to pills, but also surgical interventions or devices, as it was also mentioned yesterday. And this is particularly important today, where there is a very large development of device-based interventions for psychiatric conditions. So, for example, in this study that also was mentioned yesterday of deep brain stimulation, patients with resistant depression were assigned to 6 months of either active or some subcallosal DBS. And this was followed by 6 months of open-label DBS. And as you can see here in this table, patients from both groups improved significantly compared to baseline, but there were no significant differences between the 2 groups. And for this reason, DBS has not yet been approved by the FDA for depression, even though it's been approved for OCD or Parkinson's disease, as we all know. Now, what is the placebo effect? That's one of the main questions of this workshop, and how does it work from a clinical neuroscience perspective? Well, as it's been mentioned already, most of what we know about the placebo effect comes from the field of placebo analgesia. And in summary, classical theories of the placebo effect have consistently argued that placebo effects results from either positive expectancies regarding the potential beneficial effects of a drug, or classical conditioning, where the pairing of a neutral stimulus, in this case the placebo pill, with an unconditioned stimulus, in this case the active drug, results in a conditioned response. Now, more recently, theories of the placebo effect have used computational models to predict placebo effects. And these theories posit that individuals update their expectancies as new sensory evidence is accumulated by signaling the mismatch between what is expected and what it is perceived. And this information is then used to refine future expectancies. Now, these conceptual models have been incorporated into a trial-by-trial manipulation of both expectancies of pain relief and pain sensory experience. And this has rapidly advanced our understanding of the neural and molecular mechanisms of placebo analgesia. And so, for example, in these meta-analytic studies using these experiments, they have revealed really two patterns of distinct activations with decreases in brain activity in regions involved in brain processing, such as the dorsal medial prefrontal cortex, the amygdala, and the thalamus, and increases in brain activity in regions involved in affective appraisals, such as the BMPFC, the nucleus accumbens, and the PAG. Now, what happens in depression? Well, in the field of antidepressant placebo effects, the long-term dynamics of mood and antidepressant responses have not allowed us to have such trial-by-trial manipulation of expectancies. And so instead, researchers have used broad brain changes in the context of a randomized controlled trial or a placebo lead-in phase, which has, to some extent, limited the progress of the field. Now, despite these methodological limitations of these studies, they provide important insights about the neural correlates of antidepressant placebo effects. In particular, from these two early on studies, we can see that placebo was associated with increased activations broadly in cortical regions and decreased activations in subcortical regions. And the activations in subcortical regions were actually larger in patients who were assigned to the NSSRI treatment. We also demonstrated that very similar to pain, antidepressant placebo effects were associated with enhanced endogenous opioid release during placebo administration, predicting the response to open-label treatment after 10 weeks. And we and others have demonstrated that increased connectivity between the salience network and the rostral anterior cingulate during antidepressant placebo effects can actually predict short-term and long-term placebo effects. Now, an important limitation, and as I already mentioned, is that these studies, it is basically the delay mechanism of action of common antidepressants and the slow dynamics of mood, which really limit the possibility of accurately manipulating antidepressant expectancies. So to address this important gap, we developed a trial-by-trial manipulation of antidepressant expectancies to be used inside of the scanner. And the purpose was really to be able to further dissociate expectancy and mood dynamics during antidepressant placebo effects. And so the basic structure of this task involves an expectancy condition where subjects are presented with a 4-second infusion cue, followed by an expectancy rating cue, and a reinforcement condition, which consists of 20 seconds of sub-neurofeedback, followed by a mood rating cue. Now, the expectancy and the reinforcement condition map onto the classical theories of the placebo effect that I explained earlier. During the expectancy condition, the antidepressant infusions are compared to periods of calibration where no drug is administered. And during the reinforcement condition, on the other hand, some neurofeedback of positive sign 80% of the time is compared to some neurofeedback of baseline sign 80% of the time. And so this 2-by-2 study design results in 4 different conditions: the antidepressant reinforced, the antidepressant not reinforced, the calibration reinforced, and the calibration not reinforced. And so the cover story is that we tell participants that we're testing the effects of a new fast-acting antidepressant compared to a conventional antidepressant, but in reality, they are both saline. And then we tell them that they will receive multiple infusions of these drugs inside of the scanner while we record their brain activity, which we call neurofeedback. So then patients learn that positive neurofeedback compared to baseline is more likely to cause mood improvement, but they're not told that the neurofeedback is simulated. Then we place an intravenous line for the administration of the saline infusion, and we bring them inside of the scanner. For these kind of experiments, we recruit individuals who are 18 through 55 with or without anxiety disorders and have a Hamilton Depression Rating Scale greater than 16, consistent with moderate depression. They are antidepressant medication-free for at least 21 days, and then we use consenting procedures that involve authorized deception. Now, as suspected, behavioral results during this task consistently show that antidepressant expectancies are higher during the antidepressant infusions compared to the calibration, especially when they are reinforced by positive some neurofeedback. Now, mood responses also are significantly higher during positive sign neurofeedback compared to baseline. But this is also enhanced during the administration of the antidepressant infusions. Now, interestingly, these effects are moderated by depression severity, such that the effects of the task conditions on the expectancies and mood ratings are weaker in more severe depression, even though their overall expectancies are higher and their overall mood are lower. Now, at a neural level, what we see is that the presentation of the infusion cue is associated with an increased activation in the occipital cortex and the dorsal attention network, suggesting greater attention processing engaged during the presentation of the treatment cue. And similarly, the reinforcement condition revealed increased activations in the dorsal attention network with additional responses in the ventral striatum, suggesting that individuals process the some positive neurofeedback cue as rewarding. Now, an important question for us was, now that we can manipulate acute antidepressant placebo responses, can we use this experiment to understand the mechanisms implicated in short-term and long-term antidepressant placebo effects? And so, as I mentioned earlier, there was emerging evidence suggesting that placebo analgesia could be explained by computational models, in particular reinforcement learning. And so we tested the hypothesis that antidepressant placebo effects could be explained by similar models. So as you know, under these theories, learning occurs when an experienced outcome differs from what is expected, and this is called the prediction error. And then the expected value of the next possible outcome is updated with a portion of this prediction error as reflected in this Q learning rule. Now, in the context of our experiment, model predicted expectancies for each of the 4 trial conditions would be updated every time the antidepressant or the calibration infusion cue is presented, and an outcome, whether positive or baseline neurofeedback, is observed based on a similar learning rule. Now, this basic model was then compared against 2 alternative models. One, which included differential learning rates to account for the possibility that learning would depend on whether participants were updating expectancies for the placebo or the calibration, and then an additional model to account for the possibility that subjects were incorporating positive mood responses as mood rewards. And then finally, we constructed this additional model to allow the possibility of the combination of models 2 and 3. And so using Bayesian model comparison, we found that the model 4, which included a placebo-biased learning and a reinforcement by mood, dominated all the other alternatives after correction for Bayesian omnibus risk. Now, we then mapped the expected value and reward prediction error signals from our reinforcement learning models into our BAL data. And what we found was that expected value signals map onto the salience network, BAL responses, whereas reward prediction errors map onto the dorsal attention network BAL responses. And so altogether, the combination of our model-free and model-based results revealed that the processing of the antidepressant infusion cue increased activation in the dorsal attention network, whereas the encoding of the expectancies took place in the salience network once salience had been attributed to the cue. And then furthermore, we demonstrated that the reinforcement learning model predicted expectancies encoded in the salience network trigger mood changes that are perceived as reward signals. And then these mood reward signals further reinforce antidepressant expectancies through the formation of expectancy mood dynamics defined by models of reinforcement learning, an idea that could possibly contribute to the formation of long-lasting antidepressant placebo effects. And so the second aim of my care work was really going to look at these, in particular, how to use behavioral neural responses of placebo effects to predict long-term placebo effects in the context of a clinical trial. And so our hypothesis was that during placebo administration, greater saline attribution to the contextual cue in the salience network would transfer to regions involved in mood regulation to induce mood changes. And so in particular, we hypothesized that the DMN would play a key role in belief-induced mood regulation. And why the DMN? Well, we knew that activity in the rostral anterior cingulate, which is a key node of the DMN, is a robust predictor of mood responses to both active antidepressant and placebos, implying its involvement in non-specific treatment response mechanisms. We also knew that the rostral anterior cingulate is a robust predictor of placebo analgesia, consistent with its role in cognitive appraisal, prediction, and evaluation. And we also had evidence that the SN to DMN functional connectivity appears to be a predictor of placebo and antidepressant responses over 10 weeks of treatment. And so in our clinical trial, which you can see the concert diagram here, we randomized 6 individuals to placebo or escitalopram 20 milligrams. And this table is just to say that there were no significant differences between the 2 groups in regard to the gender, race, age, or depression severity. But what we found interesting is that there were also no significant differences in the correct belief assignment, with 60% of subjects in each group approximately guessing that they were receiving escitalopram. Now, as you can see here, participants show lower MADRS scores at 8 weeks in both groups, but there was no significant differences between the 2 groups. However, when splitting the 2 groups by the last drug assignment belief, subjects with a drug assignment belief improved significantly compared to those with a placebo assignment belief. And so the next question was, can we use neuroimaging to predict these responses? And what we found was that at neural level, during the expectancy process, the salience network had an increased pattern of functional connectivity with the DMN, as well as with other regions of the brain stem, including the thalamus. Now, at the end, we also found that increased SN to DMN functional connectivity predicted expectancy ratings during the antidepressant placebo fMRI task, such that higher connectivity was associated with greater modulation of the task conditions on expectancy ratings. Now, we also found that enhanced functional connectivity between the SN and the DMN predicted the response to 8 weeks of treatment, especially among individuals who believed that they were on the antidepressant group. Now, this data supports that during placebo administration, greater salient attributions to the contextual cue is encoded in the salience network, whereas belief-induced mood regulation is associated with an increased functional connectivity between the SN and the DMN. And altogether, this data suggests that enhanced SN to DMN connectivity enables the switch from greater salient attribution to the treatment cue to DMN-mediated mood regulation. And so finally, and this is going to be brief, but the next question for us was, can we modulate these networks to actually enhance placebo-related activity? And in particular, we decided to use theta burst stimulations, which can potentiate or depotentiate brain activity in response to brief periods of stimulation. And so in this study, participants undergo 3 counterbalance sessions of TBS with either continuous, intermittent, or sham, known to depotentiate, potentiate, and have no effect. And so each TBS is followed by an fMRI session during the antidepressant placebo effect test, which happens approximately an hour after stimulation. The inclusion criteria are very similar to all of our other studies. And our pattern of stimulation is pretty straightforward. We do 2 blocks of TBS. And during the first block, stimulation intensity is gradually escalated in 5% increments in order to enhance tolerability. And during the second session, the stimulation is maintained constant at 80% of the motor threshold. Then we use a modified CTBS session consisting of 3 stimuli applied at intervals of 30 Hertz with bursts repeated at 6 Hertz for a total of 600 stimuli in a continuous strain of 33.3 seconds. And then the ITBS session consists of a burst of 3 stimuli applied at intervals of 50 Hertz with bursts repeated at 5 Hertz for a total of 600 stimuli during 192 seconds. We also use a sham condition where 50% of subjects are assigned to sham TBS, simulating the ITBS stimulus pattern, and 50% are assigned to sham TBS, simulating the CTBS pattern. Now, our target is the DMN, which is the cortical target for the dorsal medial, the cortical target for the DMPFC, which is the cortical target for the DMN. And this corresponds to the, and we found these effects based on the results from the antidepressant placebo fMRI test. And so this target corresponds to our heuristic scalp, which is located 30% of the distance from the nation to the Indian forward from the vertex and 5% left, which corresponds to an EEG location of F1, and the connectivity map of this region is actually results in activation of the DMN. Now, we can also show here the E-field map of this target, which basically demonstrates supports a nice coverage of the DMN. And so what we found here is that ITVS compared to sham and CTVS enhances the effect of the reinforcement condition on mood responses. And we also found that at a neuro level, ITVS compared to CTVS show significant greater ball responses during expectancy processing within the DMN, with sham responses in the middle, but really not significantly different from ITVS. Now, increased ball responses in the ventromedial prefrontal cortex were associated with a greater effect of the task conditions of mood responses. And so altogether, our results suggest that first trial by trial modulation of antidepressant expectancies effectively dissociates expectancy mood dynamics. Antidepressant expectancies are predicted by models of reinforcement learning and are encoded in the salience network. We also showed that enhanced ascent to DMN connectivity enables the switch from greater salient attribution to the treatment cue to DMN-mediated mood regulation, contributing to the formation of acute expectancy mood interactions and long-term antidepressant placebo effects. And ITVS potentiation of the DMN enhances placebo-induced mood responses and expectancy processing. And so with this, I would just like to thank my collaborators that started this work with me at the University of Michigan, but mostly the people in my lab and all of my collaborators at the University of Pittsburgh, as well as the funding agencies.
Wonderful presentation, really terrific way of trying to disentangle a different mechanism in placebo response in depression, which is not an easy feat. So there are no specific questions in the Q&A. I would encourage everybody attending the workshop to please post your question to the Q&A. Every panelist can answer in writing, and then we will answer more questions during the discussion. But please don't hesitate. I think I will move on to the next speaker. We have only a couple of minutes, so we're just gonna move on to Dr. Schmidt. Thank you so much. We can see your slides. We cannot hear you.
Can you hear me now?
Yes, thank you.
Thank you. So I'm Liane Schmidt. I'm an INSAM researcher and team leader at the Paris Brain Institute. And I'm working on placebo effects, but understanding the appetitive side of placebo effects. And what I mean by that, I try to explain you in the next couple of slides.
Video?
Yes.
Please turn on your video.
Sorry?
Could you please turn on your video, Dr. Schmidt?
Yes. Sorry. Yeah. Here I am. Okay, great. Sorry.
Thank you.
Sorry about that. So it's about the appetitive side of placebo effects, because actually placebo effects on cognitive processes such as motivation and biases and belief updating, because these processes actually play also a role when patients respond to treatment and when we measure placebo effects, basically when placebo effects matter in the clinical setting. And this is done at the Paris Brain Institute, and I'm working also in collaboration with the Pitié-Salpêtrière Hospital's psychiatry department to get access to patients with depression, for example. So my talk will be organized around 3 parts. On the first part, I will show you some data about appetitive placebo effects on tastelessness, hunger sensations, and reward learning. And this will make the bridge to the second part, where I will show you some evidence for asymmetrical learning biases that are more tied to reward learning and that could contribute actually or can emerge after fast-acting antidepressant treatment effects in depression. And why is this important? I will try to link these two different parts, first part, second part, in a third part, to elaborate some perspectives on the synergies between expectations, expectation updating through learning mechanisms, motivational processes, and drug experiences, and which we might harness actually by using computational models, such as, for example, Wagner models, as Marta just showed you also evidence for this in her work. The appetitive side of placebo effects is actually known very well from a field of research in consumer psychology and marketing research, where price labels, for example, or quality labels can affect decision-making processes and also experiences like taste pleasantness experience. And since we are in France, one of the most salient examples for these kind of effects comes from wine tasting. And many studies have shown that basically the price of wine can influence how pleasant it tastes. And we and other people have shown that this is mediated by activation in what is called the brain valuation system, so regions that encode expected and experienced reward. And one of the most prominent hubs in this brain valuation system is the ventral medial prefrontal cortex, that what you see here on the SPM on the slide, that can explain how that basically translates these price label effects on taste, pleasantness, liking. And what is interesting is also that its sensitivity to monetary reward, for example, obtaining by surprise a monetary reward, it activates basically, the VMPFC activates when you obtain such a reward, surprisingly. And the more in participants who activate the VMPFC more in these kind of positive surprises, these are also the participants in which the VMPFC encoded more strongly the difference between expensive and cheap wines, which makes a nice parallel to what we know from placebo hypoalgesia, where it has also been shown that the sensitivity of the reward system in the brain can moderate placebo analgesia with participants with higher reward sensitivity in the ventral striatum, for example, another region, showing stronger placebo analgesia. So this is to basically hope that you appreciate that these effects parallel nicely what we know from placebo effects in the pain domain and also in disease. So we went further beyond actually, so beyond just taste liking, which is basically experiencing rewards such as wine. But what could be, could placebos also affect motivational processes per se? So when we, for example, want something more. And one way to study is to study basic motivations, such as, for example, hunger, that are long sought actually, and eating behavior that is long conceptualized to be driven by homeostatic markers, hormonal markers such as ghrelin and leptin that signal satiety and energy stores. And as a function of these different hormonal markers in our blood, we're going to go and look for food and eat food. But we also know from the placebo effects on taste pleasantness that there is the possibility that our higher order beliefs about our internal states, not our hormones, can influence whether we want to eat food, whether we engage in these types of very basic motivations. And that we tested that and other people also, that's a replication in the study where we gave healthy participants who came into the lab in the fasted state a glass of water. And we told them, well, water sometimes can stimulate hunger by stimulating the receptors in your mouth. And sometimes you can also drink a glass of water to kill your hunger. And a third group, a control group, was given the glass of water, telling them it's just water, it does nothing on hunger. And then we asked them to rate how hungry they feel over the course of the experiment. And it's a 3-hour experiment, everybody is fasted. And they have to do this food choice task in an fMRI scanner, so everybody gets hungry over these 3 hours. But what was interesting, and that what you see here on this rain cloud plot, is that participants who believed or who drank the water suggested to be a hunger killer increased in their hunger ratings less than participants who believed the water will enhance their hunger. So this is a nice replication, what we already know from the field. Other people have shown this too. And the interesting thing is that it also affected this food wanting, this motivational process, how much you want to eat food. So when people laid in the fMRI scanner, they saw different food items and they were asked whether they want to eat it or not for real at the end of the experiment. So it's incentive compatible. And what you see here is basically what we call stimulus value. So how much do you want to eat this food? And the hunger sensation ratings that I just showed you before parallel what we find here, that people in the decreased hunger suggestion group wanted to eat the food less than in the decreased hunger suggestion group, showing that this is not only an effect on subjective self-reports or how you feel your bodily signals about hunger, it's also about what you would actually prefer, what your subjective preference of food that is influenced by the placebo manipulation. And it is also influencing how your brain valuation system again encodes the value of food, your food preference. And that's what you see on this slide 2. You see the ventromedial prefrontal cortex, the yellow boxes, that the more yellow they are, the stronger they correlate to your food wanting. And you see on the right side with the temporal time courses of the VMPFC that food wanting encoding is stronger when people were on the increased hunger suggestion group than in the decreased hunger suggestion group. So basically what I've showed you here is 3 placebo effects. Placebo effects on subjective hunger ratings, placebo effects on food choices, and placebo effects on how the brain encodes food preference and food choices. You could wonder, these are readouts, so these are behavior readouts, neural readouts, but you could wonder what is the mechanism behind, basically, what is in between the placebo intervention and basically the behavior and neural readout of this effect. And one snippet of the answer to this question is when you look at the expectation ratings. For example, expectations have long been shown to be one of the mediators, the cognitive mediators of placebo effects across domains. And that's what we see here too, especially in the hunger killer suggestion group. The participants who believed that the hunger, that the drug will kill their hunger more strongly were also those whose hunger increased less over the course of the experience. And this moderated activity in a region that you see here, which is called the medial prefrontal cortex, that basically activated when people saw food on the screen and thought about whether they want to eat it or not. This region activated, but its activity was positively moderated by the strength of the expectancy about the glass of water to decrease their hunger. So the more you expect that the water will decrease your hunger, the more the MPFC activates when you see food on the screen. It's an interesting brain region because it's right between the ventromedial prefrontal cortex that encodes the value, the food preference, and the dorsolateral prefrontal cortex. And it has been shown by past research to connect to the VMPFC when participants self-control, especially during food decision-making paradigms. But another mechanism or another way to answer the question about the mechanism of how the placebo intervention can affect these behavior and neural effects is to use computational modelings to better understand the preference formation basically. And one way is drift diffusion modelings. So these drift diffusion models come from perceptual research for understanding perception, and they are recently also used to better understand preference formation. And they assume that your preference for a yes or no food choice, for example, is a noisy accumulation of evidence. And there are 2 types of evidence you accumulate in these decision-making paradigms. It's basically how tasty and how healthy food is. How much you like the taste, how much you consider the health. And this could influence the slope of your evidence accumulation, how rapidly basically you reach a threshold towards a yes or no. It could also be that the placebo manipulation could influence the slope. But the model allows to test several other hypotheses. It could be that the placebo intervention basically affected also just the threshold, like that reflects how carefully you made the decision towards a yes or no choice. It could be your initial bias, that is basically initially you were biased towards a yes or no response, or it could be the non-decision time, which reflects more sensory-motor integration. And the answer to this question is basically that 3 parameters were influenced by the placebo manipulation, basically how much you integrated healthiness and tastiness in your initial starting bias. So you paid more attention to the healthiness when you believed that you were on a hunger killer, and more to the tastiness when you believed that you were on a hunger enhancer. And similarly, you were initially biased towards accepting food more when participants believed they were on a hunger enhancer than on a hunger killer. Interestingly, so this basically shows that the decision-making process is biased by the placebo intervention, and basically also how much you filter information that is most relevant. When you are hungry, basically taste is very relevant for your choices. When you are believing you are less hungry, then you have more actually space, or you pay less attention to taste, but you can also pay attention more to healthiness of food. And so the example that shows that this might be a filtering of expectation relevant information is to use psychophysiological interaction analyses that look basically at the brain activity in the VMPFC, that's our seed region. Where in the brain does it connect when participants see food on a computer screen and have to think about whether they want to eat this food or not? And what we observed that it connected to the DLPFC, the dorsolateral prefrontal cortex region, and it's a region of interest that we localized first to be sure it is actually a region that is activating during interference resolution. Basically, when we have to filter information that is most relevant to a task in a separate Stroop localizer task. So the VMPFC connects stronger to the DLPFC interference resolution region, and this is moderated especially in the decreased hunger suggestion group by how much participants considered the healthiness against the tastiness of food. To wrap this part up is basically that we replicated findings from previous studies about appetitive placebo effects by showing that expectancies about the efficiency of a drink can affect hunger sensations, how participants make and form their food preferences, make food choices, and value encoding in the ventromedial prefrontal cortex. But we also provided evidence for underlying neurocognitive mechanisms that involve the medial prefrontal cortex that is moderated by the strength of the hunger expectation, that the food choice formation is biased in a form of attentional filtering mechanism toward expectancy congruent information, that is taste for an increased hunger suggestion group and healthiness for the decreased hunger suggestion group. And this is implemented by regions that are linked to interference resolution, but also to valuation, preference encoding. So why should we care? In the real world, it is not very relevant to provide people with deceptive information about hunger influencing ingredients of drinks. But studies like this one provide insights into cognitive mechanisms of beliefs about internal states and how these beliefs can affect the interoceptive sensations and also associated motivations, such as economic choices, for example. And this can actually also give us insights into the synergy between drug experiences and outcome expectations that could be harnessed via motivational processes. So translated basically via motivational processes. And through it, maybe lead us to better understand active treatment susceptibility. And I'm going to elaborate on this in the next part of the talk by going a little bit far, coming a little bit far, not talking about or showing evidence about placebo effects, but yes, before that, yes. So basically, it is linked. So these motivational processes have long been suggested actually to be also part of placebo effects or mechanisms of placebo effect, and that's called the placebo reward hypothesis. And it's based on findings in Parkinson's disease that have shown that when you give Parkinson patients a placebo but tell them it's a dopaminergic drug, well, you can measure dopamine in the brain, and the dopamine, especially the marker for dopamine, its binding potential decreases. That's what you see here on these PET scan results, and that suggests that the brain must have released endogenous dopamine. And dopamine is very important for expectations and learning, basically learning from reward, and clinical benefit is a kind of reward that patients expect. So it is possible that basically when a patient expects reward, clinical benefit, it's their brain releases dopamine in reward-related regions such as the VMPFC or the ventral striatum. And we have shown this in the past that the behavior consequence of such endogenous dopamine release under placebo could be linked to reward learning indeed. What we know is that, for example, that Parkinson patients have a deficit in learning from reward when they are off dopaminergic medication, but this normalizes when they're under active dopaminergic medication. So we wondered if, based on these PET studies, under placebo, the brain releases dopamine. Does this also has a behavior consequences on their reward learning ability? And that's what you see here on the screen, on the right side of the screen, is that the Parkinson patients basically tested on placebo show similar reward learning abilities as under active drug. And this again was also underpinned by increased correlation of the ventromedial prefrontal cortex, again, this hub of the brain evaluation system, to the learned reward value that was stronger in the placebo and active drug condition compared to baseline off drug condition. And I want to make now this link to another type of disease where also motivation is deficitary, and which is depression. And depression is known to be maintained or is hypnotized to be maintained by this triad of very negative beliefs about the world, the future, and oneself, which is very insensitive to belief disconfirming information, especially if the belief disconfirming information is positive, so favorable. And this has been shown by cognitive neuroscience studies to be reflected by a sort of lack of good news, bad news bias or optimism biases in belief updating in depression. This good news, bad news bias is basically a bias healthy people have to consider favorable information that contradicts initial negative beliefs more than negative information. And this is healthy because it avoids a worsening of beliefs. And it also encodes a form of motivational process because good news have a motivational salience. So it should be more motivating to update beliefs about the future, especially if these beliefs are negative, when we learn that our beliefs are way too negative and get information about that this confirms this initial belief. But depressed patients, they lack this, this good news, bad news bias. So we wonder what happens when patients respond to antidepressant treatments that give immediate sensory evidence about being on an antidepressant. And these new fast-acting antidepressants, such as ketamine, are these types of antidepressants that patients know right away whether they got the treatment through dissociative experiences. And so could it be that this affects also the cognitive model of depression? So this was the main question of the study. And then we wondered again, what is the computational mechanism? And is it linked again also, as shown in the previous studies, to reward learning mechanisms, so biased updating of beliefs? Is it linked to clinical antidepressant effects and also potentially outcome expectations, which makes the link to placebo effects? So patients were given were performing a belief updating task 3 times before receiving ketamine infusions, and then after a first infusion, and then 1 week after a third infusion. At each testing time, we measured the depression with the Montgomery-Åsberg depression rating scale, and patients performed this belief updating task where they were presented with different negative life events, like, for example, getting a disease, losing a wallet here, for example, and they were asked to estimate their probability of experiencing this life event in the near future. Then they were presented with evidence about the contingencies of this event in the general population, what we call the base rate. And then they had the possibility to update their belief, knowing now the base rate. And this is, for example, a good news trial where participants initially overestimated the chance for losing a wallet and then learns it's much less frequent than initially thought, and updates, for example, 15%. And in the bad news trials, you initially underestimated your probability of experiencing this adverse life event. And if you have a good news, bad news bias, well, you're going to consider this information to a lesser degree than in a good news trial. And that's exactly what happens in the healthy controls that you see on the leftmost part of the screen. I don't know whether you can see the mouse, but basically, so you have belief updating on y-axis, and this is the healthy age-matched controls to the patients. And you can see here updating after good news, updating after bad news. We tested the participants, this participant group, also 2 times within a week. You can see that bias, there is a bias that decreases a little bit with sequential testing in the healthy controls. But importantly, in the patients, the bias is there also before ketamine treatment, but it becomes much more stronger after ketamine treatment. It emerged basically. So patients become more optimistically biased after ketamine treatment. And this correlates to the MADRS scores. Patients who improve more with treatment are also those who show a stronger good news, bad news bias after 1 week of treatment. And now we wondered again about the computational mechanisms. So one way to get at this using a Rescola-Wagner model, a reward reinforcement learning model that basically assumes that updating is proportional to your surprise, what is called the estimation error, the difference between the initial estimate and the base rate. And this is weighed by a learning rate. And the important thing here is that the learning rate has got 2 components, a scaling parameter and an asymmetry parameter. And the asymmetry parameter basically weighs in how much the learning rate varies after good news, after positive estimation error than after negative estimation errors. And what we can see that in healthy controls, there is a stronger learning rate for positive estimation errors, and it is less stronger for negative estimation errors, translating this good news, bad news bias. It's basically an asymmetrical learning mechanism. And in the patients, the asymmetrical learning is non-asymmetrical before ketamine treatment, and it becomes asymmetrical as reflected in the learning rates after ketamine treatment. So what we take from that is basically that ketamine induced an optimism bias. But an interesting question is whether basically what comes first? Is it basically the improvement in the depression that we measured with the Montgomery-Åsberg depression rating scale, or is it the optimism bias that emerged and that triggered basically, since it's a correlation, we don't know what comes first. And an interesting side effect or side we put in the supplement was that in 16 patients, it's a very low sample size, the expectancy about getting better also correlated to the clinical improvement after ketamine treatment. So we have 2 expectancy ratings here about the efficiency about ketamine and also what patients expect their intensity of depression will be after ketamine treatment. So that suggests that the clinical benefit is kind of in part or synergistically seems to interact with the drug experience that emerges, that generates an optimism bias. And to test this more, we continued data collection just on the expectancy ratings and basically wondered how the clinical improvement after first infusion links to the clinical improvement after third infusion. And we know from here that patients who improve after first infusion are also those who improved after third infusion. But is it mediated by their expectancy about the ketamine treatment? And that's what we indeed found is that basically the more patients expected to get better, the more also they got better after 1 week of treatment, but it mediated this link between the first drug experience and the later drug experiences, and suggests that there might not be an additive effect, as other panelist members today already said, put forward. It might be a synergetic link. And one way to get at these synergies is basically, again, use computational models. And this idea has been around also yesterday, that basically there could be self-fulfilling prophecies that could contribute to the treatment responsiveness and treatment adherence. And these self-fulfilling prophecies are biased asymmetrically learning mechanisms that are more biased when you have positive treatment experiences, initial positive treatment experiences, and then might contribute how you adhere to the treatment in the long term and also how much you benefit from it in the long term. So it needs both a drug experience and an expectancy. And so This is a non-published work where we played with this idea, basically using a reinforcement learning model. This is also very inspired by what we know from placebo hypoalgesia. Thor and Leonie Coben, they have a paper on showing that self-fulfilling prophecies can be harnessed with biased Bayesian and reinforcement learning models. And the idea of these models is that there are two learning rates, alpha plus and alpha minus, and these learning rates weight in differently into the updating of your expectation after drug experience.
You have 1 minute remaining.
Okay, yeah, I'm almost done. So weight differently on these drug experiences and expectations as a function of whether the initial experience was congruent to your expectations, so a positive experience, then a negative one. And here are some simulations of this model showing this basically that your expectation is getting more updated the more biased, positively biased you are. Than when you're negatively biased. And these are some predictions of the model concerning depression improvement. To wrap this up, the conclusion about this is that there seems to be asymmetrical learning that can capture self-fulfilling prophecies and could be a mechanism that translates expectations and drug experiences potentially across domains, from placebo hypoalgesia to antidepressant treatment responsiveness. And the open questions is obviously to challenge these predictions of these models more with empirical data in pain, but also in mood disorders, as Marta does, and as we do also currently at Salpêtrière, where we test the neurocognitive mechanisms of belief updating biases in depression with fMRI and these mathematical models. And this has a direct clinical implication because it could help us to better understand how these fast-acting antidepressants work and what makes it that the patients adhere to them and get responsive to them. Thank you for your attention. We are the Control Interception Attention team, and thank you also to our funders.
Fantastic presentation. Thank you so much. Without further ado, let's move on to the next speaker, Dr. Greg Corder.
Did that work? Is it showing?
Thank you.
Cool. Awesome. All right, 1 second. Let me just move so there's screen. Perfect. All right. Hey, everyone. My name is Greg Porter. I'm an assistant professor at the University of Pennsylvania. I guess I get to be the final scientific speaker in this session, what's been an amazing 2-day event. So thank you to the organizers for also having me get the honor of representing the entire field of preclinical placebo research as well. And so I'm going to give a bit of an overview, some of my friends and colleagues over the last few years, and then tell you a bit about how we're leveraging a lot of current neuroscience technologies to really identify the cell types and circuits building from the human fMRI literature that's really honed in on these key circuits for expectations, belief systems, as well as endogenous anti-nociceptive systems, in particular opioid cell types. And so the work I'm going to show from my lab is really been driven by these two amazing scientists, Dr. Blake Kimmy, a postdoc in the lab, as well as Lindsay Edjo, who recently last week just received her DSPAN F99K00 on placebo circuitry. And we think this might be one of the first NIH-funded animal projects on placebo. So congratulations, Lindsay, if you're listening. Okay, so why use animals, right? We've heard an amazing set of stories really nailing down the specific circuits in humans, leveraging MRI, fMRI, EEG, and PET imaging that give us this really nice roadmap and idea of how beliefs in analgesia might be encoded within different brain circuits and how those might change over times with different types of Bayesian modeling or updating of different experiences. And we love this literature. We in the lab, we read it in depth as best as we can, and we use this as a roadmap in our animal studies because we can take advantage of animal models that really allow us to dive deep into the very specific circuits using techniques like that are on the screen here from RNA sequencing, electrophysiology, really showing that those functional connectivity measurements in fMRI are truly existent with the axons projecting from one region to another. And then we can manipulate those connections and projections using things like optogenetics and chemogenetics that allow us really tight temporal coupling to turn cells on and off. And we can see the effects of that intervention in real time on animal behavior. And that's really the tricky part, is we don't get to ask the animals, do you feel pain? Do you feel less pain? It's hard to give verbal suggestions to animals. And so we have to rely on a lot of different tricks and really get into the heads of what it's like to be a small prey animal, existing in a world with a lot of large monster human beings around them. So we really have to be very careful about how we design our experiments. And it's hard. Placebo in animals is not an easy subject to get into. And this is reflected in the fact that, as far as we can tell, there's only 24 published studies to date on placebo analgesia in animal models. However, I think this is an excellent opportunity now to really take advantage of what's been the golden age of neuroscience technologies exploding in the last 10, 15 years, to revisit a lot of these open questions about when are opioids released? Are they released? Can animals have expectations? Can they have something like a belief structure and violations of those expectations that lead to different types of prediction errors that can be encoded in different neural circuits? So we have a chance to really do that. But I think the most critical first thing is how do we begin to behaviorally model placebo in these preclinical models? So I want to touch on a couple things from some of my colleagues. So on the left here, this is a graph that's been shown on several different presentations over the past 2 days from Benedetti, using these tourniquet pain models, where you can provide pharmacological conditioning with an analgesic drug like morphine to increase this pain tolerance. And then if it is covertly switched out for saline, you can see that there is an elevation in that pain tolerance reflective of something like a placebo analgesic response overall. And this is sensitive to naloxone, the mu opioid receptor antagonist, suggesting endogenous opioids are indeed involved in this type of a placebo-like response. And my colleague, Dr. Matt Bangart at UCSD, has basically done a fantastic job of recapitulating this exact model in mice, where you can basically use morphine and other analgesics to condition them. And so if I just kind of dive in a little bit into Matt's model here, you can have a mouse that will sit on a noxious hot plate. It's an environment that's unpleasant. You can have contextual cues, like different types of patterns on the wall. And you can test the pain behavior responses, like how much does the animal flick and flinch and lick and bite and protect itself to the noxious hot plate. And then you can switch the contextual cues, provide an analgesic drug like morphine, see reductions in those pain behaviors, and then do the same thing in the Benedetti studies. You switch out the morphine for saline, but you keep the contextual cues. So the animal has effectively created a belief that when I am in this environment, when I'm in this doctor's office, I'm going to receive something that is going to reduce my perceptions of pain. And indeed, Matt sees a quite robust effect here, where this sort of placebo response shows this elevated Paul withdrawal latency, indicating that there is endogenous antinociception occurring with this protocol. And it happens, again, pretty robustly. I mean, most of the animals going through this conditioning protocol demonstrate this type of antinociceptive behavioral response. So this is a perfect example of how we can leverage what we learned from human studies into rodent studies for acute pain. And this is also really great to probe the effects of placebo in chronic neuropathic pain models. And so here, this is Dr. Damien Borman, who was with Professor Kevin Key in Australia, now with Lauren Martin in Toronto. And here, Damien really amped up the contextual cues here. So this is an animal who has had injury to the sciatic nerve with this chronic constriction injury. So now this animal is experiencing something like a tonic chronic neuropathic pain state. And then once you let the pain develop, you can have the animals enter into this sort of placebo pharmacological conditioning paradigm, where animals will go onto these thermal plates, either hot or cool, in these rooms that have a large amount of visual, tactile, as well as odorant cues. And they're paired with either morphine or a controlled saline. Again, the morphine is switched for saline on that last day. And what Damien has observed is that in a subset of the animals, about 30%, you can have these responder populations that show decreased pain behavior, which we interpret as something like analgesia overall. So overall, you can use these types of pharmacological conditionings for both acute and chronic pain. So now what we're going to do in our lab is it's a bit different, and I'm really curious to hear the field's thoughts because everything I'm about to show is completely unpublished. Here we're going to use an experimenter-free, drug-free paradigm of instrumental conditioning to instill something like a placebo effect. And so this is what Blake and Lindsay have been working on since about 2020. And this is our setup in one of our behavior rooms here. Our apparatus is this tiny little device down here, and everything else are all the computers and optogenetics and calcium imaging techniques that we use to record the activity of what's going on inside the mouse's brain. But simply, this is just 2 hot plates that we can control the temperature of, and we allow a mouse to freely explore this apparatus. And we can, with a series of cameras and tracking devices, plot the place preference of an animal within the apparatus. And we can also record with high-speed videography these highly conserved sort of protective recuperative pain-like behaviors that we think are indicative of the negative affect pain. And so let me walk you through our little model here real quick. Okay, so we call this the placebo analgesia conditioning assay, or PAC assay. So here's our 2-plate apparatus here. So plate number 1, plate number 2, and the animal can always explore whichever plate it wants. It's never restricted to one side. And so we have a habituation day, let the animal familiarize itself, like, oh, this is a nice office. I don't know what's about to happen. And then we have a pretest. And in this pretest, importantly, we make both of these plates, both environments, a noxious 45 degrees centigrade. So this will allow the animal to form an initial expectation that the entire environment is noxious and it's going to hurt. So both sides are noxious. Then for our conditioning, this is where we actually make one side of the chamber non-noxious. So it's just room temperature, but we keep one side noxious. So now there's a new expectation for the animal that it learns that it can instrumentally move its body from one side to the other side to avoid and escape feeling pain. And so we'll do this over 3 days, twice per day. And then on our post-test or placebo day, we make both environments hot again. So now we'll start the animal off over here, and the animals will get to freely choose. Do they want to go to the side that they expect should be non-noxious, or what happens? So what happens? Actually, if we just look at the place preference for this, over the course of conditioning, we can see that the animals will unsurprisingly choose the environment that's non-noxious, and they spend 100% of their time there, basically. But when we flip the plates, or flip the conditions such that everything is noxious on the post-test day, the animals will still spend a significant amount of time on the expected analgesia sign. So I'm going to show you some videos here now, and you're all going to become mouse pain behavior experts by the end of this. So what I'm going to show you are both side-by-side examples of conditioned and unconditioned animals, and try to follow along with me as you can see what the effect looks like. So on this post-test day, oh gosh, let's see if this is gonna, here we go. All right, so on the top, we have the control animal running back and forth. The bottom is our conditioned animal. And you'll notice we start the animal over here, and it's going to go to the side it expects it to not hurt. Notice the posture of the animals. This animal is sitting very calm. It's putting its entire body down on the hot plate. This animal, posture up, tail up, it's running around a little bit frantically. You'll notice it start to lick and bite and shake its paws. This animal down here might have a couple little flinches, so it's letting you know that some nociception is still getting into the nervous system overall. But over the course of this 3-minute test, the animals will greatly choose to spend more time over here. And if we start to quantify these types of behaviors that the animals are doing in both conditions, what we find is that there's actually a pretty significant reduction in these nociceptive behaviors, but it's not across the entire duration of this placebo day or post-test day. So this trial is 3 minutes long. And what we see is that this anti-nociceptive and preference choice only exists for about the first 90 seconds of this assay. So this is when the video I just showed, the animal goes to the placebo side, it spends a lot of its time there, does not seem to be displaying pain-like behaviors. And then around 90 seconds, the animal, it's almost like the belief or the expectation breaks. And at some point, the animal realizes, oh no, this is actually quite hot. It starts to then run around and starts to show some of the more typical nociceptive-like behaviors. And we really like this design because this is really, really amenable to doing different types of calcium imaging, electrophysiology, optogenetics, because now we have a really tight timeline that we can observe the changing of neural dynamics at speeds that we can correlate with some type of behavior. Okay, so what are those circuits that we're interested in overall that could be related to this form of placebo? Again, we like to use the human findings as a wonderful roadmap, and Tor has demonstrated, and many other people have demonstrated, this interconnected distributed network involving prefrontal cortex, nucleus accumbens, insula, thalamus, as well as the periaqueductal gray. And so today I'm going to talk about just the periaqueductal gray because there's evidence that there's also a release of endogenous opioids within this system here. And so we tend to think that the placebo process and the encoding, whatever that is the placebo itself is likely not encoded in the PAG. The PAG is kind of the end of the road. It's the thing that gets turned on during placebo and we think is driving the antinociceptive or analgesic effects of the placebo itself. So the PAG, for anyone who's not as familiar, we like it because it's conserved across species. When we look at it in a mouse, there's one in a human, so potentially it's really good for translational studies as well. It has a very storied past where it's been demonstrated that the PAG sub-architecture has these beautiful anterior to posterior columns that if you electrically stimulate different parts of PAG, you can produce active versus passive coping mechanisms, as well as analgesia that's dependent on opioids, as well as endocannabinoids. And then the PAG is highly connected, both from ascending nociception from the spinal cord, as well as descending control systems from prefrontal cortex, as well as the amygdala. So with regard to opioid analgesia, if you microinfuse morphine into the posterior part of the PAG, you can produce an analgesic effect in rodents that is across the entire body. So it's super robust analgesia from this very specific part of the PAG. If you look at the PAG back there and you do some of these techniques to look for histological indications that the mu-opioid receptor is there, it is indeed there. There's a large amount of mu-opioid receptors. It's OPRM1, and it's largely on glutamatergic neurons, so the excitatory cells, not the inhibitory cells. They are on some of them. And as far as ePhys data goes as well, we can see that the mu-opioid receptor is there. So damgoat's an opioid agonist. We can see activation of inhibitory GERK currents in those cells. So the system is wired up for placebo analgesia to happen in that location. Okay, so how are we actually going to start to tease this out? By finding these cells, where they go throughout the brain, and then understanding their dynamics during placebo and pain. So last year, we teamed up with Carl Diceross lab at Stanford to develop a new toolkit that leverages the genetics of the mu-opioid system, in particular, the promoter for the mu-opioid receptor. And we were able to take the genetic sequence for this promoter and package it into adeno-associated viruses, along with a range of different tools that allow us to turn on or turn off cells or record their activity. And so we can use this mu-opioid receptor promoter to gain genetic access throughout the brain or the nervous system for where these mu-opioid receptors are, and we can do so with high fidelity. This is just an example of our mu-opioid virus in the central amygdala, which is a highly mu-opioid specific area. But so Blake used this tool, using the promoter, to drive a range of different transgenes within the periaqueductal gray. And right here, this is the GCAMP. So this is a calcium indicator that allows us to, in real time, assess the calcium activity of PAG mu-opioid cells. And so what Blake did was he took a mouse and he recorded the nociceptive responses within that cell type and found that the mu-opioid cell types are actually nociceptive. They respond to pain, and they do so with increasing activity to stronger and stronger and more salient and intense noxious stimuli. So these cells are actually nociceptive. And if we look at a ramping hot plate, we can see that those same mu-opioid cell types in the PAG increase their activity as this temperature on this hot plate increases. Those cells can decrease that activity if we infuse morphine. Unsurprisingly, they express the mu opioid receptor, and they are indeed sensitive to morphine. But if we give naltrexone to block the mu opioid receptors, we can see greater increased activity to noxious stimuli, suggesting that there could be an opioid tone or some type of an endogenous opioid system that's keeping this system in check, that it's repressing its activity. So when we block it, we actually enhance that activity. So it's going to be really important here. And the activity of these mu opioid PAG cells correlates with affective measures of pain. When the animals are licking, biting, and shaking its foot, when it wants to escape away from noxious stimuli, that's when we see activity within those cells. So this is just correlating different types of behavior when we see peak amplitudes within those cell types. So let me skip that real quick. Okay, so we have this ability to look and peek into the activity of mu opioid cell types. So let's go back to that placebo assay, our PAC assay that I mentioned before. If we record from the PAG on that post-test day in an animal that has not undergone conditioning, so this is when the plates are both sides are super hot, we see a lot of nociceptive activity in these cells. Here, they're bouncing up and down. But if we look at the activity of the nociception in an animal undergoing placebo, what we see is there's a suppression of neural activity within that first 90 seconds. And this actually does seem to extinguish within the later 90 seconds. So kind of tracks along with the behavior of those animals. When they're showing anti-nociceptive behavior, that's when those cells are quiet. When the pain behavior comes back, that's when those cell types are ramping up. But what about the opioids too? So the mu opioid receptor cell types, decreasing activity. What about the opioids themselves here? So the way to do this in animals has been to use microdialysis. Fantastic technique, but it's got some limitations to it. So this is a way of sampling peptides in real time and then using liquid chromatography to tell if a protein was present. However, the sampling rate is about 10 minutes. And in terms of the brain processing, 10 minutes might as well be an eternity. If we're talking about milliseconds here. But we want to know what these cells here in these red dots are doing. These are the enkephalnergic cells in the PAG. And so we needed a revolution in technologies. And one of those came several years ago from Dr. Lin Tian, who developed some of the first sensors for dopamine. Some of you may have heard of it. It's called D-Lite. And so this is a version of D-Lite, but it's actually an enkephalin opioid sensor. And so what Lin did to genetically engineer this is to take the delta opioid receptor, highly selective for enkephalin, and then link it with this GFP molecule here, such that when enkephalin binds to the sensor, it will fluoresce. And we can capture that fluorescence with microscopes that we implant over the PAG. And we can see when enkephalin is being released with sub-second resolution. And so what we did for that is we want to see if enkephalin is indeed being released onto those mu opioid receptor expressing pain encoding neurons in the PAG. And so what I showed you before is that those PAG neurons, they ramp up their activity as nociception increases. So this is a mouse standing on a hot plate. We see that nociception ramp up. What do y'all think happened with the opioids? It wasn't what we expected. It actually drops. So what we can tell is that there is a basal opioid tone within the PAG, but that as nociception increases, acute nociception, we see a decreased suppression of opioid peptide release. We think this has to do with stuff that that Tor has published on previously, that the PAG is more likely involved in updating prediction errors. And this acute pain phenomena, we think, is reflective of the need to experience pain, to update your priors about feeling pain, and to bias the selection of appropriate behaviors, like affect-related things, to avoid pain. However, what happens in our placebo assay? We actually see the opposite. So if we condition animals to expect pain relief within that PAC assay, we actually see an increase from the delta light sensor, suggesting that there is an increase in enkephalin release post-conditioning. So there can be differential control of the opioid system within this brain region. And so this next part is the fun thing you can do with animals. What if we just bypassed the need to do the placebo assay? If we know that we just need to cause release of enkephalin within the PAG to produce pain relief, we could just directly do that with optogenetics. And so we use this animal that allows us to put a red light sensitive opsin protein into the encephalinergic interneurons of the PAG. And when we shine red light on top of these cells, they turn on and they start to release their neurotransmitters. These are GABAergic and encephalinergic. So they're dumping out GABA and now they're dumping out enkephalin into the PAG. And we can visualize that using the delta light sensor from Lentien. And so here's an example of optogenetically released enkephalin within the PAG over 10 minutes. The weird thing that we still don't fully understand is that this signal continues after the optogenetic stimulation. And so can we harness the placebo effect? In mice, at least it seems we can. So if we turn on these cells strongly, cause them to release enkephalin, and then put animals back on these ramping hot plate tests, we don't see any changes in the latency to detect pain, but we see specific ablation or reductions in these affective motivational pain-like behaviors overall.
Hi, Doctor. You have 1 minute remaining.
Absolutely. Cool. All right. So in this last minute here too, just to say, I know that there's a lot of people that are always skeptical. Can we actually test, these higher order cognitive processes in animals? And if for anyone who's not a behavioral preclinical neuroscientist, you might not be aware that there's an absolute revolution happening in behavior with the use of deep learning modules that can precisely and accurately quantify animal behavior. And so this is an example of a deep learning tracking system we've built called the Light Automated Pain Evaluator that can capture a range of different pain-related behaviors, fully automated without human intervention whatsoever, that can be paired with brain recording techniques like calcium imaging that allow us to fit a lot of different computational models to understand what the activity of single neurons might be doing, let's say in the cingulate cortex, that might be driving that placebo response. And we can really start to tie now in at single cell resolution the activity of prefrontal cortex to drive these placebo effects and see if that alters anti-nociceptive behavior endogenously. So I'll stop there and thank all of the amazing people, Blake, Greg, and Lindsay, who did this work, as well as all of our funders and the numerous collaborators who have helped us do this. So thank you.
Terrific talk. Thank you so much. Really blown away. And now I'll leave the discussion to our two moderators. They're going to gather some of the questions from the chat and some of their own questions for all the presenters from today and from yesterday as well. Okay.
Should I start?
Go ahead.
Okay. Matt, you start gathering the questions. I got permission to say a few moments of comments. I wanted to say this was fantastic. I actually learned amazing amount of things. The amount of light that was brought forward about what we know about placebos and how we can possibly control placebo effects, how we can possibly harness placebo effects. There was so much light and new information. What I want to do in my 4 minutes of comments is look to the future. And I want to say, and what I mean by that is that we had many, and I want to just give my comments and you can take them or leave them, but I got a few minutes. What I want to say is that we got the lights, but we didn't put them together. And I don't, there's no way we could have. We needed to be more in the same room. How does this fit in with your model? It's hard to do. What I mean by putting things together is, I give an example. In terms of how do we control placebo effects in clinical trials? I not infrequently get asked by pharmaceutical industry, can you look at our placebo data? We just blew it. Placebo was as good as, or was almost as good as the drug. And the first thing I say is I want to talk to experts in that disease. I want to know the natural history. I want to know how you made your entry criteria so I can understand regression to the mean. I want to know what's the relationship of the objective markers and subjective markers so I can begin to think about how much is the placebo response. I always tell them I don't know. If I knew how to increase the difference between drug and placebo, I'd be a rich man, I wouldn't be an academic. And what I usually wind up saying is, get a new drug. And they pay me pretty well for that. And the reason is that they don't know anything about natural history. We're trying to harness something. And I just want to say, I've done a lot of natural history controls, and That's more interesting than the rest of the experiments because they're unbelievable, the amount of improvement people show entering a trial without any treatment. So I just want to say we need to look at other things besides the placebo effect if we want to control the placebo response in a randomized controlled trial. So I want to say that to go forward. I also want to say that we need a little bit of darkness. We need to be able to say, you know, I disagree with you. I think there's other data. And one of the things I've learned in being in placebo research is that there's a paper that contradicts your paper real quickly, and there's lots of contradictory information. It's very easy to say you're wrong. And we don't say it enough. I just want to pick one example. Please forgive me. I know that my research could be said, Ted, you're wrong, but I just want to say something. Consistently in this 2 days of talk, everyone talks about the increase of the placebo response over time. No one refers to the article published in 2022 in BMJ. First author was Mark Stone, and senior author was Irving Kirsch, and they analyzed all FDA. Mark Stone is in the division of psychiatry at Cedar at the FDA. They analyzed all data of placebo controlled trials in major depressive disorder. They had over 230 trials, way more than 70,000 patients, and they analyzed the trend over time from 1979 to the present at the publication. There was no increase in the placebo effect. There was, you know, are they right or other people right? But nothing is 100% clear right now, and we need to be able to contradict each other when we get together personally and say, I don't think that's right. Maybe that's right. I think that would help us. And the last thing I want to say is that some things are missing from the conference that we need to include in the future. We need to have ethics. Placebo is about ethics. If you're a placebo researcher in placebo controls and trials, that's an important question. What are we talking about in terms of compromising ethics? There was no discussion of that. We didn't have time, but in the future, let's do that. And the last thing I would say is we need to ask patients what their experience is. I got to say, I've been around for a long time, but the first time I started asking patients what their experiences were when they were on double-blind placebo or open-label placebo, I did it way after, you know, they finished the trial was over, and I actually took notes and went back and talked to people. They told me things I didn't even know about, and we need to have that in conferences. So what I want to say, just one more line, is I feel so much healthier because I'm an older person, and I feel this younger crowd here is significantly younger than me. Maybe Matt and I are the same age, I don't know. But I think this is really one of the best conferences I've ever went to. It was real clear data. We need to do lots of other things in the future. So with that, Matt, feed me some questions.
Okay. Thanks, Ted. I didn't realize that you were also 35, but okay. If you don't mind, I'm going to start with a question of mine. The recent emergence of intravenous ketamine for resistant depression has introduced an interesting methodologic approach that we have not seen in a long time, and that is the active placebo. So where the early trials just used saline, more recently we see the benzodiazepine midazolam while not mimicking really the full dissociative effect that many people get from ketamine. The idea is for people to feel some kind of buzz, so that they might believe that they're on some active compound and not just saline. And I wonder if the panel has any thoughts about the merits of using an active placebo, and is that something that the field should be looking into more?
I'm going to say something. Irving Kirsch published a meta-analysis of 8 studies that used atropine as a control in the depression studies, and he felt that that made it difficult to detect the placebo drug difference. But another meta-analysis said that was not true. That was common in the 80s. People started thinking about that, but I have no idea how to answer your question.
No, well, hi, Mike Detke. I think it's a great question, and you know, and I think in the presentations yesterday about devices, Dr. Listenby, I think, was talking about the ideal sham, and I think it's kind of, you know, it's very similar. The ideal active placebo would have, you know, none of the efficacy of the drug, but would of the drug in question, but would have, you know, exactly the same side effects and all other features. And of course, that's attractive, and but of course we probably never have a drug that's exactly like that. I think midazolam was a great thing to try with ketamine. It's still not exactly the same. But I'd also add that you know, it's not black and white. It's not like we need to do this with ketamine and ignore it for all of our other drugs. All of our drugs have side effects. Arguably, if you do really big chunks like classes of the relatively modern antidepressants, antipsychotics, and the psychostimulants, those are in order of bigger effect sizes in clinical trials, psychostimulants versus antipsychotics versus, and they're also in the order of roughly, I would argue that of unblinding, of functional unblinding, and of in terms of more magnitude, you know, Zyprexa will make you hungry, and also speed of onset of some of the adverse effects in stimulants and some of the type 2 or the second generation and beyond antipsychotics are, you know, they have pretty noticeable side effects for many subjects, and relatively rapidly. So I think those are all important features to consider.
Dr. Schmidt, I see the hand up, and then Dr. Reif also.
Yes, I think using like midazolam could give like some sensory sensation so that patients actually can say there's some effect on the body like immediately. But this raises actually a question whether these dissociations we observe in some patients after ketamine infusions really have a functional role, play a role for the antidepressant response. And this is still an open question. So I don't have the answer to that question. So, and I think midazolam doesn't really induce dissociations. I don't know, maybe you can isolate then the dissociations you get on the ketamine, but maybe even patients might be educated expecting some dissociative experiences, and then you destroy it basically when they don't have, so they might interpret the midazolam experience as something negative. So yeah, these self-fulfilling prophecies might again play in.
I'm just gonna add for 5 seconds, because I run a pretty large ketamine clinic, and we know very little about the effect of placebo on maintaining an antidepressant response. All the dissociation often wears off over time, and this is completely separate from the antidepressant effect. We don't have long-term placebo studies. We study a lot of the studies are extremely short-lived, and we study the acute effect, but we don't know how to sustain, how to maintain, what's the role of placebo effect in long-term treatment. So that's another field that would be really open to investigations. Dr. Reif.
Yes, following up on the issue of active placebos, I just want to mention that we did a study and comparing active placebos to passive placebos, and showing that active placebos are really more powerful. And I think the really disappointing part of this news is that it questions the blinding of our typical RCTs, comparing antidepressants versus placebos, because many patients who are in the active group, are in the drug group, they perceive these onset effects, and this will further boost the placebo mechanisms in the drug group that are not existing in the passive placebo group. So this is a challenge that further questions the validity of our typical RCTs.
Martin?
Yeah, just a quick follow-up to what Christina was saying too, is that we need to clarify whether we want to find an active control for the dissociative effects or for the antidepressant effects. And I think the approach will be very different. And this applies to ketamine, but also to psychedelics, because we were having this discussion as well. And so when thinking about how to control for or how to blind, or how we just, these treatments are very complicated. They have multiple effects. We just need to have the discussion of what are we trying to blind, because the mechanism of action of the blinding drug will be very different.
Okay.
Can I just say something about blinding? People should, there's Osbjorn Robertson, who was the author of the 1990, 1993, his New England Journal paper saying that there's no placebo effect as a myth. In 2022, published in BMJ, the largest he called it a mega meta-analysis on blinding. And he took 144 randomized controlled trials that included non-blinded evidence on the drug versus blinded evidence of drug. I'm not going to tell you the conclusion because it's unbelievable, but you should read it because it really influences it would influence what we think about blinding. That study was just recently replicated on a different set of patients with procedures in JAMA surgery 3 months ago. And blinding like placebo is more complicated than we think. That's what I wanted to say.
There's another clinical factor that's come up during our discussion has been the relationship of the patient to the provider. That we saw data showing that a warm relationship seemed to enhance therapeutic response, I believe, to most interventions. And I wonder what the panel thinks about the rise, on the one hand, of shortened clinical visits now that, for example, antidepressants are mostly given by busy primary care physicians and not specialists, and the so-called med check is a really kind of quickie visit. And especially since the pandemic, the rise of telehealth, where a person might not ever even meet their provider in person. And is it possible we're on our way to where a clinical trial could involve, say, mailing medication every week to a patient, having them do their weekly ratings online, and eliminating a provider altogether, and just looking at the pharmacologic effect? And is that that probably isn't how we want to actually treat people clinically, but in terms of research, say early phase efficacy, is there merit to that kind of approach?
Question.
Can I reply and comment on this, Dr. Rutherford?
Please.
We are very interested to see how the telemedicine or virtual reality can affect placebo effects. And we are modeling in the lab placebo effects induced via, in-person interaction versus an avatar in virtual reality. And actually, we found placebo effects with both the settings. However, when we look at empathy, the avatar doesn't elicit any empathy in the relationship. We truly need this in-person connection to have empathy. So that suggests there are outcomes that are affected by, having in-person versus telemedicine / remote interactions, but yet the placebo effects persist in both the settings. The empathy is differently modulated. And the empathy mediates, at least in our data, placebo effects only in the in-person interactions. So there is still value in telemedicine. Effects that bypass empathy completely and competence.
Dr. Hall.
You know, several of the large studies, like the Women's Health Study, Physicians Health Study, and more recently Vital, they did exactly that, where they mail these pill packs. And the population obviously is clinicians, so they're very well kind of trained and well behaved. But they follow them for years, but there's very little contact with the providers. And these are, you still have these giant, I don't know if you can call them placebo effects, but certainly many of these trials have not proven to be more effective, the drugs that they're studying, than placebo.
Dr. Atlas?
Thank you. I just wanted to chime in briefly on this important question. I think that the data that I presented yesterday in terms of first impressions of providers is relevant for this, because it suggests that even when we use things like ZocDoc to select physicians and we have headshots, that really we're making these decisions about who to see based on these kinds of just first impressions and facial features. And having the actual interactions with providers is critical for sort of getting beyond that kind of factor that may drive selection. So I think if we have situations where there's reduced chances to interact, first of all, people are bringing expectations to the table based on what they know about the provider, and then you don't really have the chance to build on that without the actual kind of therapeutic alliance. So that's why I think, even though our study was done in a artificial setting, it really does show how we make these choices when there are bios for physicians and things available for patients to select from. So I think there is a really important expectation being brought to the table before the treatment even occurs.
Okay, thanks. Dr. Lisenby.
So thank you for raising this great question, Matt. And I have a little bit of a different take on it. Equity in access to mental health care is a challenge. And the more that we can leverage technology to provide and extend the reach of mental health care, the better. And so telemedicine and telepsychiatry, we've been thrust into this era by the pandemic, but it existed before the pandemic as well. And it's not just about telepsychotherapy or teleprescription monitoring pharmacotherapy, but digital remote neuromodulation is also a thing now. There are neuromodulation interventions that can be done at home that are being studied. And so there have been trials on transcranial direct current stimulation at home with remote monitoring. There are challenges in those studies differentiating between active and sham. But I think you're right, Matt. We may have to rethink, how do we control remote studies when the intensity of the clinician contact is very different. But I do think that we should explore these technologies so that we can extend the reach and extend access to research and to care for people who are not able to come into a typical research lab setting.
Can I add something on this, because it's fun and it's also criticizing myself? In 2008, I did this very nice study showing you can increase the doctor-patient relationship, and as you increase it, the placebo effect got bigger and bigger, like a dose response. A team in Korea that I worked with replicated that and just published that replication. The replication came out with the exact opposite results. The less doctor-patient relationship, the less intrusive, the less empathy, got better effects. We're dealing with very complicated, culturally constructed issues. And I just want to put it out there. The sand is soft. And I'm really glad someone contradicted a major study that I did.
Exactly. The cultural component is so critical. What we observe in one context, in one country, but even within the same in-group or out-group, can be completely different in Japan, in China, or somewhere else, in South America, South Africa. So we need larger studies and more across-country collaborations.
Dr. Schmidt.
I just wanted to raise a point, not really like it's more like a comment. Like, there's also very interesting research going on in the interactions between humans and robots. And usually humans treat robots very badly. And so I wonder what could be like, here we focus on very human traits like empathy, competence, like what we look in. But when it happens to artificial intelligence, for example, and when we have to interact with algorithms, basically, like all these social interactions might completely turn out completely different, actually, and also have different effects on placebo effects. Just a thought.
Dr. Reif.
Well, yesterday I expressed a belief for showing more warmth and competence, but I will modify it a little bit today because I think the real truth became quite visible today, and that is that there is an interaction between these non-specific placebo effects and the drug effects. In many cases, at least, we don't know whether there are exceptions from this rule, but in many cases we have an interaction. And to learn about the interaction means that we need study designs that do not only modulate drug intake versus placebo intake, but they also modulate the placebo mechanisms, the expectation mechanisms, the context of the treatment. And only if we have these 2x2 designs modulating drug intake and modulating context and psychological factors, then we learn about the interaction. You cannot learn about the interaction if you modulate only one factor. And therefore, I think what Luana and others have said, that a drug can be quite powerful and effective in one context, but maybe even misleading in another context. I think this is true, and we have to learn more about that. And all the studies that have shown from basic science to application that there could be an interaction, they are all indicating this line and this, to this necessity that we use more complex designs to learn about the interaction.
Yes, and you know the rodent studies we've seen, I think, have a powerful message for us just in terms of being able to control a lot of variables that are just totally beyond our control in our usual human studies. You know, I mean, it always seemed to me that, for example, if you're doing just an antidepressant versus placebo trial in patients, well, you know, for some people, going into the clinic once a week to get ratings, that might be the only day of the week that they get up and take a shower, get dressed, have somebody ask them how they're doing, you know, I mean, that have some human interaction. And so showing up for your Hamilton rating could be a therapeutic intervention that, of course, we usually don't account for in the pharmacotherapy trial. Yeah, the number of variables really can escalate in a hurry when we look at our trials closely.
Dora wants to say something.
Please.
Oh yeah, thanks, Ted. So I just wanted to add on to the interaction issue, which came up yesterday, and which Winfried and others just commented on, because it seems like it's really a crux issue. If the psychosocial or expectation effects and other things like that are entangled with specific effects, so that one can influence the other and they might interact, then yeah, we need more studies that independently manipulate specific drug or device effects and other kinds of psychological effects independently. And I think I wanted to bring this back up again because this is an idea that's been out here for a long time. I think the first review on this was in the 70s, like 76 or something. And it hasn't really been picked up for a couple of reasons. One is it's hard to do the studies. But second, when I've talked to people who are in industry and pharma, they are very concerned about changing the study designs at all for FDA approval. And since we have some, FDA and regulatory, perspectives here yesterday, I wanted to bring that up and see what people think, because I think that's been a big obstacle. And if it is, then that may be something that would be great for NIH to fund instead of pharma companies, because then, there's a whole space of drug psychological or device, neurostimulation psychological interactions that could be explored.
Okay. We also had a question that yesterday there was discussion in a naloxone trial about sex differences in placebo response, and wonder if there are any further thoughts on studies of sex differences or diversity in general in placebo trials. Yes.
Luana, thank you.
Yes.
So we definitely see sex differences in placebo effect. And I show also how, for example, women respond to arginine vasopressin in a way that we don't observe in men. But also you ask about diversity. Currently, actually a paper got just about accepted today where we look at where people live in the Maryland state, and even the location where they are based make a difference in placebo effects. So people who live in the most, distressed area of the greatest Baltimore area tend to have lower placebo effects as compared to a not distressed population. And we define that on a series of criteria, not merely ethnicity, race, but we take into account education, the occupation they have, the income, and so on. So it is interesting because across studies, consistently, we see an impact of diversity, And in that sense, I echo Lisanbay's comment that we need to find a way to reach out to these people, and truly improve access and opportunity for diversity. Thank you for asking.
Well, thank you. Another issue that that came up yesterday had to do with the pharmacogenomics, and there was a question or a question/comment about using candidate approaches, and aren't those problematic? And I wonder if there are any
What approaches?
Candidate genes.
Catherine, address that.
Yeah, well, I think that we have to start where we are, and there's, I think the psychiatric field has had a really tough time with genetics, right? They've invested a lot and sadly don't have as much to show for it as they would like to, and I think that has really tainted this quest for genetic markers of placebo and really to study these interaction factors. But it's really important to not, I think, use that to stop us from going forward and looking to identify what's there, because I think when you start to scratch the surface, there are interactions. You can see them, they're replete in the literature. And what's really fascinating is everybody who finds them they don't see them when they report their study. And even some of these vasopressin studies, not obviously Luana yours, but I was reading one the other day where they had seen tremendous differences by genetics in response to arginine vasopressin, and they interpreted it they totally ignored what they were seeing in placebo and talked about who responds to drug. And so I think that not only do we need to start looking for what's happening, we need to start being more open-minded and paying attention to what we're seeing in the placebo arm and accounting for that, taking that into account to understand what we're seeing across a trial in total.
I wanted to take a second to comment on also patient selection and trying to figure out, depending on the site, who are the patients who went a treatment-resistant depression clinical trial. If we eliminate from the discussion professional patient and we think about the patients who are more desperate, patients who don't have access to care, patients who are more likely to have psychosocial stressors, or the other extreme, there are patients who are highly educated, they go on clinicaltrials.gov and they search out, but they're certainly not representative of the general populations we see in a clinical setting. They're somewhat different. And then if you think about the psychedelics trial, you go from 5,000 patients applying for a study in the study ended up recruiting 20, 30. So they're absolutely not representative of the general population we see in terms of diversity, in terms of comorbidities, in terms of psychosocial situations. So there's another factor that adds to the complexity of differentiating what happens in a clinical setting versus an artificial setting like a research study. Tor?
No, the question of who enters trials, and I think the larger issue of diagnosis in general has I think really been a challenge to the field for many years. Ted and I go back a ways, and just looking at depression, which of course, has, I think, dominated a lot of our discussion these last couple of days, with good reason. If now I realize the good database, my understanding is that the good database of placebo-controlled trials go back to the late 90s, is what we heard yesterday. And if you go back further, the tricyclic era not only dealt with different medications, which we don't want to go back to, but if you think about practice patterns, then on the one hand, the tricyclics, most non-specialists steered clear of. They required a lot of hands-on. They required titration slowly up. They had some concerning toxicities. And so it was typical that psychiatrists would prescribe them, but family docs would not. And that also had the effect of a kind of naturalistic screening. That is, usually people would have to reach a certain level of severity before they were referred to a psychiatrist to get a prescription for medication. And so more mildly ill people either wound up probably inappropriately on tranquilizers or no treatment at all, and moderately to severely ill people wound up on tricyclics. And of course, inpatient stays were more common in those days, which again was another kind of screening. So it was the sort of thing. I mean, in the old days, I heard people talk about, well, you could, if you go to the inpatient ward, you could easily collect people to be in your clinical trial, and you kind of knew that they were vetted already, that they had severe depression. The general sense was that the placebo response would be low, though there's no real evidence for that. But the thing is, once we had the SSRIs, on the one hand, the market vastly expanded because they're considered more broad spectrum. People with milder illness and anxiety disorders now are appropriate candidates, and they're easier to dispense. The concern about overdose is much less, and so they're mostly prescribed by non-specialists. And so it's the sort of thing where we've seen a lot of large clinical trials where it doesn't take much to reach the threshold of for entry. I mean, if I go way back, and this is just one of my personal concerns over many years, the finer criteria, which I think were the first good set of diagnostic criteria based on data, based on the literature, those which were published in 1972, to have a diagnosis of major depression called for 4 weeks of symptoms, or actually literally, I think it said 1 month. And DSM-3 came out in 1980, and it called for 2 weeks of symptoms. And I don't know, I've not been able to find any documentation of how the 1 month went to 2 weeks, except that the DSM, of course, is the manual that's used in clinical practice. And you can understand, well, you might not want to have a too high a bar to treat people who are seeking help. But I think one of the challenges of DSM is it was not meant as a research manual, though that's often how it's used. So ever since that time, those 2 weeks have gotten reified. And so my point is, it doesn't take much to reach diagnostic criteria for DSM, now 5-TR, major depression. And so if someone is doing a clinical trial of an antidepressant, it is tempting to enroll people who honestly meet those criteria, but the criteria are not very strict. And so, I wonder whether that, contributes to the larger placebo effect that we see today. And end of soapbox. The question, I'd like to revisit an excellent point that Dr. Lisenby raised yesterday, which has to do with the research domain criteria, the RDoC criteria. And I don't know if anyone on the panel has had experience in using that in any trials, and whether you see any merit there. Could RDoC criteria essentially enrich the usual DSM type clinical criteria in terms of trying to more finely differentiate subtypes of depression who might respond differently to different treatments?
So Matt, I think Tor has been very patient in having his hand up. So maybe before we go to the next question, I don't know, Tor, if you had some comment from previous discussion.
Oh, sure, thanks. I think I just wanted to make a comment on the candidate gene issue. And I think it links to what you were just saying as well, Dr. Rudorfer, in a sense. But I think it relates to the issue of predicting individual differences in placebo effects and using that to enhance clinical trials, which has been really sort of a difficult issue. And in genetics, I think what's happened, as many of us know, is that there were many findings on particular candidate genes, especially COMT and other sort of a particular subset of genes in Science and Nature, and none of those really replicated when larger GWAS studies started being done. And I think the field of genetics really focused in on reproducibility and replicability in very large sample sizes. That makes it, so I think my genetics colleagues tell me something like 5,000 is a minimum for even making it into their database of genetic associations. And so that makes it really difficult to study placebo effects in sample sizes like that. And at the same time, there has been this trend in psychology and in science, really in general, towards reproducibility and replicability that was probably in part sort of evoked by John Ioannidis's provocative claims that most findings are false. But there is something really there. So there's been many teams of people who have tried to pull together, like Brian Nozick's work with Open Science Foundation and many lab studies, to replicate effects in psychology, with much higher power. So that's also, there's this sort of increasing effort to pull together consortia to really test these things rigorously. And I wonder if we might not have a GWAS study of placebo effects in 100,000 people or something, which is what would convince a geneticist that there's some kind of association. And so I'm wondering what the ways forward are. And I think one way is to increasingly come together to pool studies or do larger studies that are pre-registered and even registered reports, which are reviewed before they're published, so that we can test some of these associations that have emerged in these what we call early studies of placebo effects. And I think if we pre-registered and found something in sufficiently large and diverse samples, that might make a dent in convincing the wider world that essentially that there is something that we can use going forward in clinical trials, and that pharma might be interested in, for example, as well. So that's kind of a take on that. I'm wondering what people think.
Sure, just my 2 cents. I completely agree with you. I think that the way forward is to pool our resources to look at this and not simply stop. I mean, like, I think when things don't replicate, we need to understand why they don't replicate. I think there's a taboo on looking beyond. If you've, pre-specified it and you don't see it, then it should be over. I think in at least this early stage when we're trying to understand what's happening, I think we need to allow ourselves deeper dives, not for action, but for understanding. Yeah, I think that, so I agree with you. Let's pool our resources and start looking at this. The other thing I just want to point out that's really interesting is that when we've looked at some of these clinical trials at the placebo arm, we actually learn a lot about natural history. We just did one in Alzheimer's disease. And in the placebo arm, the genome-wide significant hit was CETP, which is now a clinical target in Alzheimer's disease. So you can learn a lot by looking in the placebo arms of these studies, not just about whether or not the drug is working or how the drug is working, but what's happening in the natural history of these patients that might change the effect of the drug.
Martha, do you want to say something? You have your hand up, please.
Yes, thank you. And this is just kind of a follow-up to what everybody's saying, but I do think that the issue of inter-individual variability is very important. And I do think that, one thing that maybe kind of explains some of the things that Ted was also saying at the beginning, that there's a little bit of a lack of consistency or like a way to put all of these findings together. But the fact that we think about it as a one single placebo effect, and we do know that there's not one single placebo effect, but even within different clinical conditions, is the neurobiology, the placebo effect the same in depression as it is in pain, or are there aspects that are the same? For example, expectancy processing, but there are some other things that are very specific to the clinical condition, whether it's pain processing, mood, or some others. And so I think we face the reality, at least from the neurobiology perspective, that a lot of the research has been done in pain, and that is still there is very, very little being done, at least in psychiatry and across many other clinical conditions, that we just don't know. And we don't really even know if the placebo effect, how does the placebo effect look when you have both pain and depression, for example. And so those are still very open questions that kind of reflect our state, right? That we're making progress, but there's a lot to do.
Winfred, do you want to say something? You have your hand up. Winfred.
Yes, thanks. I want to come back to the question whether we really understand this increase of placebo effects. I don't know whether you have good explanations for that, but personally, I'm more like a scientist. I can't believe that people are nowadays more reacting to placebos than they did 20 years ago. So there might be other explanations for this effect, like we changed the trial designs. We have more control visits maybe nowadays compared to 30 years ago. But there could be also other factors like publication bias, which was maybe more frequent, more often 30 years ago than it is nowadays, with the need for privilege registration. So there are a lot of methodological issues that could explain this increase of placebo effects or of responses in the placebo groups. I would be interested whether you think that this increase is well explained, or what your explanations are for this increase.
Winfred, I want to give my opinion. I've been following this issue because I remember the first time it was reported by these scientists in Cleveland with 40 patients, 50 patients. And I said, oh my God, and the newspapers had it all over. The placebo effect is increasing. There's this boogeyman around, and everyone started believing it. I've been consistently finding as many papers saying there's I've really been collecting them. There's no change over time versus there are changes over time. And when I read the original article, I said, of course there's differences. The patients that got recruited in 1980 were different than the patients in 1990 or 2010. They were either more chronic, less chronic. They were recruited in different ways. And that's really an easy explanation of why things change. Natural history changes, people's health problems are different. And I actually think that the Stone's meta-analysis with 77 70,033 patients says it very clearly. It's a flat line from 1979 to, and the more data you have, the more you have to believe it. That's all. So that's my personal opinion. And I think we actually are very deeply influenced by the media. I mean, I can't believe this. The mystery of the placebo. We know more about placebo effects, at least as compared to many drugs on the market. It's crazy. Anyway, that's my opinion, Winfred. Thanks for letting me say it.
Thanks, Ed. We have a question for Greg. Greg, question is, I wonder what the magic of 90 seconds is. Is there a physiologic basis to the turning point when the mouse changes behavior?
Yeah, I think I had addressed it in a written post somewhere. We don't know. We see a lot of variability in those animals. So in this like putative placebo phase, some mice will remain on that condition side for 40 seconds, 45 seconds, 60 seconds, or they'll stay there the entire 3 minutes of the test. We're not exactly sure what's driving the difference in those different animals. So these are both male and females. We see this effect in both male and female C57 black 6 mice. So this is a genetically inbred animal. We try to always restrict the time of day of testing. We do reverse light testing. So this is the animal's wake cycle. But there are things like higher dominance hierarchies within the cages. So these alphas versus betas, they might have different levels of or pain thresholds. But the breaking of whatever the anti-nociceptive effect is, I mean, they're standing on a hot plate for quite a long time. So at some point, those nociceptors in the periphery are going to, become sensitized and signal. And at some point, it's to the animal's advantage, right, to pay attention to pain. You don't want to necessarily go around not paying attention to something that's potentially very dangerous or harmful to you. But we would have to scale up the number of animals substantially, I think, to really start to parse out like what the difference is that would, account for that. But it's an excellent point, though.
Carolyn.
So I want to thank all today's speakers and wonderful presentations today. I wanted to just go back for a second to Dr. Priscina's point about thinking about the placebo effect as not a monolith, and also thinking about individual disorders. And so I'm a clinical trialist and do research in obsessive compulsive disorder. And a lot of things that are written in the literature and meta-analysis, the OCD has one of the lowest placebo rates. And so, from what we gathered today, I guess to turn the question on its head is why is that the case? Why is that the case? And does that say something about OCD pathology? And what about it, right? How can we really get more refined in terms of different domains and really thinking about the placebo effect? So just want to say thank you again and to really having a lot of food for thought.
Thanks. So, as we're winding down, one of the looming questions on the table remains whether research gaps, and where do you think the next set of studies should go? And I think if anyone wants to put some ideas on the table, they'd be welcome.
It's okay. I guess I clicked a little fast. Well, one of the areas that I mentioned in my talk that is hard for industry to study, or there's a big disincentive, which is I talked about having third-party reviewers, review source documents and videos or audios of the HAM-D, MADRS, whatever, and that there's not much controlled evidence, and it's fairly simple design, within a largish controlled trial, do this with half the sites and don't do it with the other half. Blinding isn't perfect. I haven't thought about this. It probably can be improved upon a lot. But imagine you're the sponsor who's paying the $20 million in 3 years to run this clinical trial. You want to test your drug as best as you possibly can. You don't want to really be paying for, this methodology. So that might be earlier on, Tor or someone mentioned, there might be some specific areas where this might be something for NIH to consider picking up, because that methodology is being used in hundreds of trials, I think, today. The third-party remote reviewer. So there's an area to think about.
Thanks. Howie.
Yeah, Caroline just mentioned one of the gap areas, really trying to understand why some disorders are more amenable to the placebo response than others, and what can that teach us? That sounds like a research gap area to me. Also, throughout these 2 days, we've heard a number of research gap areas having to do with methodology, how to do placebos or shams, how to assess outcome, how to protect the blind, how do you select what your outcome measures should be? And then also today, my mind was going very much towards what can preclinical models teach us, and the genetics, the biology of a placebo response, the biology underlying individual differences in placebo response. There may be clues there. Carolyn, to your point, placebo response being lower in OCD, and yet there are some OCD patients who respond. What's different about them that makes them responders? And so studies that just look at within a placebo response versus non-response, or gradation response, or durability of response, and the mechanisms behind that, these are questions that I think may ultimately facilitate getting drugs and devices to market, but certainly are questions that may be helpful to answer at the research stage, particularly at the translational research stage, in order to inform the design of pivotal trials that you would ultimately do to get things to market. So it seems like there are many stages before getting to the ideal pivotal trial. So I really appreciate everyone's input, and let me stop talking because I really want to hear what Dr. Hall has to say.